Buenos días a todas y a todos. Vamos a dar inicio a nuestro segundo día de simposio, el cual hoy día va a tratar sobre los mecanismos de memoria, almacenamiento de memoria y su recuperación. Les recordamos que las preguntas deben ser realizadas a través de la opción de preguntas y respuestas que está en la parte de abajo del panel de Zoom. Y además les recordamos que hoy tendremos sesiones hasta la 1 de la tarde, terminando esta aproximadamente a las 2 de la tarde. Vamos a tener 2 recesos de aproximadamente 15 minutos durante la sesión de la mañana. ¿Okay? Voy a comenzar a hablar en inglés y voy a presentar a nuestro panelista de hoy. Today, we're gonna have our second day of symposium, and the theme of today is the mechanisms of memory store and recall. The first talk, it will be from Dr. Michele Pignatelli, and he's a research scientist at RIKEN MIT Center for Neuronal Circuit Genetics at the Picauer Institute for Learning and Memory, also from the Department of Biology and the Department of Brain and Cognitive Science at MIT. Dr. Pignatelli research is focused on the organization and development of the hippocampal circuits, and also he will talk today about the Engram cell excitability and memory recall. So we would like to thanks Michele for accepting our invitation, and please you can start when you want.
Hello, everybody. Good morning, and thanks for this kind invitation. In particular, I would like to thank Francisco for it. So let me share the screen, and we will start this presentation on the work that has been done on these Engram circuits, hippocampal Engram circuits, and their relationship to memory. So, here we are. Is it everything okay?
Yes.
Yes, perfect. So let me start. So today I will present, this was my last study, which was published in 2019, and is the relationship between the excitability state of Engram cells and the memory recall. So this is a brief outline of what I'm going to present. I will give you a brief background, basically, of the Engram labeling technology, the reactivation, the artificial retrieval, and the effect on synapses. Then I will move to the study of excitability itself, with a particular focus on the inward rectifier potassium channels, this leaky conductance that is crucial to regulate the cellular excitability. After it, I will talk about the behavioral role of this excitability with a series of behavioral correlates. One is allocation, another pattern separation and pattern completion, concluding with a causal link between this leaky conductance, which is KIF 2.1, the excitability, and all these behavioral, different behaviors. So let me begin with a definition of the Engram. Although it may seem obsolete because we are talking about something of the beginning of the 1900, basically, this theory was conceived by Richard Semon, a German zoologist. And basically, he wrote this book called Die Mneme. And in this book, basically, he defines the rules of memory traces. Okay. Sometimes Engram and memory traces are used as a synonymous, but as you will see, there's a slight difference. So the definition of an Engram is into the book is the enduring through primarily latent modification in the irritable substance produced by a stimulus. So by Engram, we mean the physical chemical change that is happening into the nervous tissue. Okay, this physical chemical change then leads to the formation of these memory traces. So which are the properties of these engrams? Number one, that learning must induce a persistent change on specific brain cells, the engram cells. These engram cells can be reactivated upon appropriate retrieval condition. So we form engram cell, and this engram cell, they are reactivated to elicit recall. The reactivation of the Engram cells elicits memory retrieval. So, as you can see, how can we test this Engram hypothesis? We need, number one, a tool that is able to label these particular cells, right? And in this sense, I will show you that we use basically the CFOS promoter as a tool to label this cell. Why CFOS? CFOS, there's two main reasons. Number one is an activity, a yearly immediate gene, and an activity-dependent gene, which is crucial for the plasticity. In fact, the knockout of FOS impairs learning and long-term potentiation. So the second part is the recall, the reactivation of the Engram cells. How can we test it? Very simple, because if we have a technology to label these cells, we can then use afterwards a CFOS antibody in response to memory recall. And then we can run an overlap index and see if this overlap is beyond chance level, or we can devise a control and compare it. Finally,
excuse me, Michele.
Yes.
Could you turn on your camera, please?
Yes, wait a second.
Thank you.
So, I need to find Zoom. Sorry, 1 second, start my video. Is it okay?
Thank you.
Yes, perfect.
It's okay.
Yes. Okay. Okay, the third part is the reactivation. In the sense, we want to be able to reactivate artificially these cells and to see if we can elicit, again, memory retrieval, right? And this we can do it by using optogenetics. So, as I told you before, the CFOS promoter is the heart of this technology. What we use is, in general, is a CFOS TTA mouse available on JAX, which was made by the Mayford lab some time ago. And we inject in the dentate gyrus, because this has been the area, the primary area of our research, TRE channel opsin to EYFP virus. So what's going on now? The TTA element, which is under the control of a FOS promoter, you can see it in the panel on the right side, basically activate the TRE element to elicit basically the production of channel opsin to EYFP. There's a particular point that the TTA can be blocked by doxycycline, which is an antibiotic that you can introduce in the diet. If a TTA is blocked, the system, it doesn't work. So this, it allows us basically to constrain the temporal window. It's not perfect. It takes 24 hours. Therefore, we have our own noise level, but nevertheless, we succeed to label these cells, as you can see in the histological panel underneath. On the right side, you can see that we label around 6 to 7% of the overall DG population. And consider that the home cage cell, so in case you do not condition the animal, is about 2 to 3%. So this is our noise level, and we succeed to deal with it. Now, let's move to the next slide, which is the reactivation. Here is a typical example of an experiment that you can run to test the reactivation hypothesis. You have two groups. They both start on day 1 in a context that we call B. On day 2, we remove the doxycycline from the diet to activate the Engram machinery, basically our construct, and we condition the animal into context A. On day 3, then, group 1 get tested in context A, and group B get tested in context B. You can see the difference. Since the recall in A elicit freezing, the one in B, it doesn't, no? And immediately after, we can sacrifice the animal, extract the brain, and run an histological analysis when we look at the overlap between the FOS that is expressed through the channel opsin YFP expressed through our virus, and the CFOS itself, which will be labeled through an antibody in red, you can see. And this is the overlap index, so also known as reactivation index, between the the AA group and the AB group. And you can see that the difference is quite striking there in terms of reactivation. So this confirms basically the hypothesis of Seman about the reactivation of the Engram system. So far we fulfill two of the criteria of Seman. Now let's go to see the third, which is the most important because we have to get a behavioral signature of a memory reactivation. And this, we do it through optogenetic, by shining light on this granule cell, Engram granule cell, that express channelrhodopsin 2. So as you can see, this is an experiment. This was done in France. It was the first time it was replicated by me during a summer school in Bordeaux. So we mounted everything. I prepared the animal. So this is the first time transatlantic engram, basically. So this is light off. You can see light on. The animal will start freezing, you see? And you will see the rebound when the light goes off again. So it's evident the conflict of behavioral states between exploration and defensive state. Light off, look at how does it move. And imagine this video has been speed up 5 times for the sake of this presentation. So how do we analyze this data? So a typical experiment that we run usually consisted in 2 groups. In this case, we use as a control an amnesic animal. We induced amnesia by injection of an isomycin, another protein synthesis inhibitor that get injected after learning, after the training session. So usually you have an habituation phase where the animal is habituated to a context, to the light in particular. So in this case, you know that by shining light without conditioning, the animal doesn't freeze. That's the first control you want to see. Then we train our animal into this red context, as you can see, and we have 2 groups, a plus shock and a minus shock, okay? A no shock control group. It's another fundamental control because you want to also be sure that all these things are not just a stimulation of a label, genetically encoded label line. Then we have a first recall test in the conditioned context. It goes back into context A for optogenetic activation, and finally another final test into the context B to test for long-term effect. So as you can see, basically the animal freezes to optogenetic activation, and look the plot C, basically, the green bar there, the green column. This is basically the saline group that has been shocked and is in context, he gets the light activation and he gets a 30% freezing. The anisomycin is around 10%. This is the amnesic animal. Okay, 1 second. So then you can see on the panel D the profile, and finally the long-term effect. There's no long-term effect of these things. The animal continue to be like this. So you can see the on-off pattern in panel D, right? For both control, the saline, and the anisomycin. So this experiment at that time was quite important because it was the first time someone succeeded to retrieve a memory from an amnesic state. So this was the important part of it. But it was just part of a message of a paper. One of the important things of this analysis was that we correlate with states with changes in synapses. And how we did it? We did it by injecting, by labeling our engram cells in red, as you can see, and then by getting us a synaptic access infecting the perforan path itself, so in the entorhinal cortex, with a Cancasian rhodopsin virus. So let me show you how does it look in a brain. This is a small schematic. This is allow you to patch an engram cell that is labeled and a non-engram cell that is not labeled, and to activate optogenetically in vitro the perforan path fibers which are labeled from the entorhinal cortex. We can have our 2 groups again, the saline and the anisomycin. You see the animal gets shocked, basically. Then the two groups, they may receive saline or anisomycin. You wait 24 hours, and then you proceed with the patch clamp analysis. And this is how does it look, an histological picture of this preparation. So you can see the two cells. The one on the right is an M-cherry positive. You see the beautiful perforant path in green there. And this is a classic experiment. Most of the cells patch are within 50 micron of distance. So this allows you to run a paired analysis. It's a kind of internal control within the same slice, and it's very convenient. And the things that you can measure are basically the AMPA-NDA ratio, which is a sign of long-term potentiation, and the dendritic spine density. And this is what you get. In terms of dendritic spines, you can see that the Engram cells of the saline group, they have higher dendritic spine, while the Engram cell of the anisomycin group and the non-Engram cell of all the groups, they have a lower. They are all at the baseline level. Analogous for the saline, for the AMPA-NDA ratio, we get exactly the same profile. Okay, so this data, they suggest that indeed the synapses are potentiated and that this potentiation is not present in the anisomycin group. I remember anisomycin blocks the protein synthesis following learning. We did one more experiment to confirm the presence of the memory trace. And in this case, we labeled the Engram mossy fibers with the STIA-rich anaerolopsin 2 YFP and the CA3 pyramidal cell in CA3 with M-cherry. Then what we did, we patched M-cherry positive and M-cherry negative CA3 pyramidal cells, and we activate the mossy fibers optogenetically, computing a probability of connection between the Engram mossy fiber, the Engram CA3 cells, and the Engram and the non-Engram CA3 cells. And what you can see is that there's a higher probability of connection between Engram mossy fiber and Engram CA3 cells in both saline and anisomycin. So in anisomycin, the trace is preserved, even if the amplitude of this connection is reduced, but the connectivity is still there. So this suggests that the substrate of the memory retrieval must be a feed-forward Engram circuit. So now that we are done with this introduction to the Engram, and so you understood the behavioral effect, the relevance, and the synaptic correlates of it, we can move to excitability. And excitability, I synthesize it in this way. Perhaps some people may find it incorrect, but in reality, this is what does it mean. It's the capability of a neuron to respond to a stimulus. So, in other words, having the same stimulus amplitude, for example, by changing the excitability of a cell, we change its reactance to the stimulus. And to be more clear, we could go back to a little bit of biophysics, but nothing special. It's basically Ohm law. And you can see here look just at the diagram, we have three examples of cells. One has 2 channels, another 4 channels, and another one has 8 channels, for example. And a different resistance of the cell that goes down progressively because the resistance and the conductance of the cell are inversely related, right? So you can see here that by injecting the same amount of current I, you see this negative square pulse I is the same to all the cells, we get a different voltage deflection down there, which is much higher for the cell with less channels. So now these channels in general are called leaky conductance. They are the most important one that controls the membrane resistance of the cells. Among the leaky conductance, and I think most of you are also they study anesthetics. For example, Francisco. The common leaky conductance are the inward rectifier, the K2P, and the HCN, which is the IH current. So the granule cells of the dentate gyrus, they mainly express the KIR 2.1. And I will show you afterward how important is this conductance later on. So just to remind you, high membrane resistance is equal to high excitability and low membrane resistance to low excitability. I focus on the membrane resistance, but our parameters of excitability are the action potential threshold and the resting membrane potential. Okay, so let's move now to a little bit of historical background. So the link between excitability and memory is not new. We can say that it was really postulated by von Neumann in the 1950s, something like this, where he assumed that a cell could, during a memory state, it could have changed state by just switching excitability state. But this, it wasn't true. It was mainly synaptic. The main nature is synaptic today. This is what is believed. But in the 1986, John Disseroff, basically, he demonstrated a change in the intrinsic excitability of a CA1 pyramidal cell in the hippocampus of a rabbit that has been conditioned to trace cycling conditioning. And the main change was basically a decrease into the post-burst hyperpolarization. In our case, we work on granule cell, which is a cell with different electrical properties. So, based on some preliminary experiment that we did, what we found, it was that this excitability, it could have been a transient phenomenon and not a persistent one. Therefore, we devised three groups. Number one, a no recall group that you can see on the top. The second one is a 5 minutes group, which is, what does it mean 5 minutes group? That the animal, it gets sacrificed 5 minutes after recall. We prepare the slices, they are incubated for an hour, and then we patch. And then we had a third group, which is a 3-hour group, where we do exactly the same thing over 5 hours. We run the recall test, we wait 3 hours in this case, we sacrifice the animal, and we proceed with a patch clamp analysis. So by comparing these three, we have a picture that is at least more clear about the situation. And also it saves you a lot of time because otherwise you become crazy, in other words, with all these groups. So in these three plots that you can see underneath, what I did, I plot the Rio base, which is the current needed to elicit a single spike, as a function of membrane resistance for the no recall group on the left, the 5 minute group on the center, and the 3-hour group on the right. So as you can see, number one, the two variables, of course, are correlated. But the interesting part, it happens in the 5 minutes group, where you see a shift of these parameters towards the high excitability state. In other words, there's a decrease in the Rio base and an increase in the membrane. So therefore, the cell, in this case, it finds itself into a high excitability state. But we also run a complete screening of this excitability state with a 5 minutes, a 10 minutes, a 30 minutes, a 1 hour, a 2 hour, a 3 hours, and a 24-hour group, in order to have the temporal profile of this transient phenomena. And what we found is that the phenomenon lasts about an hour and then it decays. Another important thing is this phenomenon is dependent of NMDA activation because injection of CPP, an NMDA blocker, before recall inhibits, as you can see, the formation of this highly excitable state. Another interesting thing that we had for our preliminary experiment and we repeated is the effect of protein synthesis inhibitors. If we inject an isomycin, again, our amnesic agent, immediately after recall, what's going on is that the cell finds itself locked, and we patch 24 hours before after. We lock the cell in a persistent, highly excitable state. So what does it mean, this? That in order to go back to a regular state, the cell needs to produce new ion channels, new leaky conductance, to go back to a low excitability state. These are the additional parameters I told you. In the top plot, you can see the action potential threshold as a function of a resting membrane potential, and there's no change in the three groups. In the middle panels, this is a classic IF curve, current versus firing frequency. And you can see that there's a nice change only and exclusively for the 5 minutes group, where the Engram cell in green display, of course, a high excitability. From the point of view of the hyperpolarization, we couldn't find any difference for hyperpolarization amplitude and duration for the three groups. So another important thing when you do this kind of experiment is to evaluate the context specificity of this phenomena. Is it really linked to the recall of a memory, or it can just be elicited by any kind of arousal stimulus, like a new context, for example? So again, here we have our control. We see that our excitability is high. This is our classic 5 minutes group. Now, what's going on if we insert, instead of making a recall in context A, we just place the animal in context C, a new context, after shock? We see no change in the excitable state of a cell. We can do again another experiment and activate these cells optogenetically in context C and then patch the cell. Even here in this case, we see no change in the excitable cell state of a cell. And this is because, as we showed before, it requires an NDA activation, probably. That's the point. So having said so, the picture that emerged from this data is the following, that in response to memory recall, we get this activation of a perforant path, we can assume, we can be quite confident about it. And this, it changed in turn through an NDA activation, it changed the excitable state of a cell. On the green bar, you can see it on the right side, which stays high for about an hour, and then it goes back to a regular state. So what is interesting now is to understand what is the behavioral significance of this transient change, so we can design experiments that target exclusively this portion where we know that we have high excitability. But before we go this, let's go a little bit to the mechanism. I told you that here 2.1 is crucial to regulate the excitability of granule cell. And here you can see a plot in current clamp and in voltage clamp there. But the important things that for you to understand the importance, you know that the leaky conductance is crucial to regulate the membrane resistance, and this is a fact. Now, if we use this ML133, which is a blocker of the KIR2 family, so very precise blocker, and we add this drug into the path, we get rid of about 68% of the overall leaky conductance. And this is very well correlated with the membrane resistance, as you can see there. So we see the impact of this ion channel on the excitability of the cell. And this is a wonderful target for manipulation later. So a mechanism, in other words. Here on the side is a panel where we tested several other leaky conductants, and you see the effect is very, very small compared to our ML133. So what's going on now at the KIR 2.1 level in the 5 minutes group? So we can run our 5 minutes experiment as usual, and then apply this drug on our slice and compare before and after. How does it go? So when we do this, you can see that in the highly excitable state, so what we can do is to measure in Engram and non-Engram cells the KIR 2.1 before and after the drug application. And then we make a subtraction of a current, and we can devise the fraction that of the contribution of KIR 2.1 during this excitable state. And you can see that in Engram cells on the right one, no, the KIR 2.1 in the high excitable state is reduced, no? So we have a reduction, we hypothesize that this reduction was due to an internalization of these channels. We test this hypothesis of internalization by doing a confocal analysis and staining the KIR 2.1 with monoclonal antibodies, and comparing the fluorescent intensity for Engram and non-Engram cells on the adjacent cells on the same focal plane. And we got that in the 5 minutes group, the overall fluorescence goes down in agreement with internalization hypothesis, as well as in the vancomycin, but not in the no recall and in the 3-hour group. So having said so, let me summarize briefly for this moment before we switch to behavior for the last 10 minutes of the talk. So the Engram cell excitability state increases in response to memory recall. This increase is transient, lasting about an hour, a bit more than an hour. The increase is NMDA dependent. The return to low excitability state is protein synthesis dependent. The main mechanism involves internalization of KIR2.1 ion channel. So, what it could be the behavioral role of this transient change? So, the first hypothesis we tested, I mean, perhaps misguided by the literature, it was the allocation. The allocation hypothesis. What is this allocation hypothesis? It postulates that cells in a high excitability state are preferentially recruited during learning. So if it's true for learning, I thought it could be true also for recall, right? It's just an hypothesis. So how do you test it? Basically, you condition the animal to context A in day 1, and on day 2, you run a recall test. 5 minutes later, you place it in B, and on day 3, you see if it freezes in B, because some of a cell, they must be common to A and B due to the allocation hypothesis. And you see why, because some of the cells in A and in B are in common. You see the overlap in yellow. This is the allocation hypothesis. So again, we have our 3 groups. Don't be scared by these panels because in reality they are very easy. We have a no recall group, the 5 minutes group, and the 3-hour group. They all get conditioned in context A on day 1. The no recall group has no recall on day 2. The 5 minutes has a recall followed by exposure to context B 5 minutes later. The 3 hours, 3 hours later. And then we test in context B and again in context A as a supplemental control. So what you can see, what we expected it was to see an enhancing freezing in context B similar to A. So the green should have been similar to the red as a high. In reality, we didn't find this thing. We find actually slightly the opposite. So we thought, based on this negative result, which at that time was a kind of tragedy, but we thought perhaps it could be instrumental for discrimination and not for allocation. Sometimes you don't want to make one memory of 2, you want to keep them segregated. There's no reason. And how do we reconcile allocation and discrimination? This is a problem, actually. So why not to test discrimination? And how do you test discrimination? By devising a parent separation task. So, and this is what we did. So, separation, it means the ability of a network to discriminate similar input patterns. Basically, in behavioral terms, it's segregation of conflicting cues. So you need similar cues in the 2 contexts. Again, what you can do is you shock your animal in context A on day 1, and then you do again the same thing with a 5 minutes, 3-hour no recall group, as usual, where you run a recall test, and then you place it in the context AB, which is the similar context, one with conflicting cues. You test on day 3 AB. So it's the same diagram as the allocation, nothing to be afraid. We have a conditioning on day 1. On day 2, the no recall group has no recall, and just directly exposed to the context with conflicting cues called AB, no? While the 5 minutes and the 3 hours, they get a recall session 5 minutes and 3 hours before, no? And on day 3, we have a test in B, and again, the control in A. So what's going on? This is the recall in A in day 2. Of course, it's only for the 5 minutes and 3 hours. You see, more or less, we get our freezing. And this is what's going on in AB. In the no recall, we have generalization, and the animal freezes as well in the 3 hours, but not in the 5 minutes group. In the 5 minutes group, the animals switch the behavior from defensive to exploratory, if I have to be more honest. If we don't want to talk about memories, this. So this is what you see in the animal. The animal explores more, and it was quite surprising to us. So this effect is gone the day after, so it's just a transient effect, no? And there's no problem in the last panel. I mean, there's a little bit of extinction, which is due to all the exposure, but everything is regular. So we can say that, yes, there's an enhancement of the discrimination capability of the animal. When we test the pattern separation, we can't avoid to test pattern completion again. And how do you test the pattern? By pattern completion, we mean the ability of a network to retrieve a full memory given only partial stimuli. So completion of partial cues, in other words. How do you test it? You use the so-called immediate shock paradigm. So this immediate shock paradigm is quite simple. You take the animal, you put it in a cage, you wait a few seconds, you shock it and you remove it. Very simple. It has been shown by Fancelo that if you do like this, the animal doesn't remember the context. It won't freeze the day after if you expose it. So what we said, what we thought, we thought just let's expose the animal today to context A without shock on day 1. Then we run a recall session on day 2, and 5 minutes, 3 hours, or 3 hours later, for example, we reintroduce it in A again, and we run an immediate shock protocol with different placement to shock interval. What does it mean? 5 seconds and 10 seconds, the time to shock, placement to shock. And the final test on day 3. So when we do this thing, again, we have our no recall and 5 minutes and 3 hour group. What we see is the following, that only in the 5 minutes group for the 5 second placement to shock interval and 10 second placement to shock interval, we have an increase in the freezing level. They succeed to complete the pattern. So having said so, I have shown no effect on allocation and an important effect on pattern separation and pattern completion. I would suggest that pattern completion in reality is really the crucial mechanism underlying the entire behavioral phenomena. How come? Because if we imagine in presence of full cues, we get this 3 granule cell activated. If now we have partial cues, for example, only 2 cells will be activated. But if a cell is in a high excitable state, even if it receives 1 input, it will still be able to fire. So overall, this will represent a kind of new mechanism of pattern completion, right? Complementary to the autoassociative mechanism, one based on recurrent collaterals of the CA3 network. And this mechanism enhances the access to memory itself. So now let's go to the manipulation, to the causal relationship. We have a behavioral correlate, we have our transient excitability, and we know that here is our mechanism that we can manipulate. So what we did in this case, we hyperexpress within the physiological range, here to 0.1 channel, in the Engram cells. So it's like we keep the excitability low, understand? And this is how does it look like. You can see that the membrane resistance in average is lower on the lower level of a control group, and same thing the real base is much higher. So we get a cell that is not highly excitable, but still functional. At the level of a consolidation, the memory still works. We have no problems to recall the memory in this case. So we can run our pattern separation and pattern completion test. So the pattern separation in this case is done with the usual protocol, in this case, just the addition of a DOX that we have to remove from the diet in order to express the key into the cell. But you can see that on day 1, we label the cell. On day 2, basically, we run the 5 minutes test, first in A and then in AB, and then day 3 and day 4 are our test day in context AB and context A. So on day 2, the recall in A, there's no change. When we place the animal in AB, you can see, you remember that in pattern separation, the level of freezing was very low, and this is what the RFP control means. But if we express here to 0.1, the freezing goes up again. No change for day 3 and day 4. For pattern completion, we do exactly the same thing, the same protocol as before, no? And what we can show is that you remember that in the 5 minutes group, the freezing went up because you succeed to complete more. If we put the key in the dentate gyrus Engram cell, the freezing goes down again. So another important, the last thing is an important control experiment. You can say, yes, but what if you label a random population instead of the Engram cells? And you can do it by exposing the animal to an unrelated context, a context B, and labeling the cell of context B. And then you run again the pattern separation and the pattern completion task. And in this case, you see there's no change. So this, it supports our conclusions. Now, one of the problem in this, this is the last minutes, this is the last slide, so don't be afraid. One of the main problem of this study is that there has been done, although it's an integrative study with multiple techniques, it has been done in vitro. So the point is that there's a time to sacrifice the animal, you have to make the slice and then wait for the slice to recover. Although we keep the slice at 23 degrees, so they are frozen basically, before they get patched at 35 degrees, the situation is not ideal. So we can do today the same experiment by using genetically encoded voltage sensors, such as Archon, in combination with some compatible optogenetic tools. And in the Adam Cohen lab, they developed this technology called OptoPatch, where you can stimulate optogenetically a cell in vivo, and you can record the voltage fluctuations microscopically. So basically, this is the concept on the left side. So what do you do in this case is that you inject a combination of this Archon, which is combined to Sheriff, which is your optogenetic actuators, and you label your Engram cell with TRE and Cherry. You implant a lens into the hippocampus, and then you can see in panel C, before learning, you get a sparse population expressing Archon. After learning, you have a tag for the Engram cells, and then you can study also after recall. And here are preliminary data where we stimulate optogenetically, and we do an IF curve, basically, or a ramp test to catch the real base. So this is actually the one current ongoing project, and is done into CA1. So probably will be a slightly different situation, but we are very excited about it. So overall, in conclusion, recall induces a transient change in the excitability state of Engram cells. During the state of excitability, the efficacy of memory retrieval is enhanced. The substrate of this form of short-term memory is the same memory trace, and not a different area, for example. These excitability states, so the low and the high, mediate different behavioral outcome, for example, defensive or exploratory, a landmark of adaptive behavior. So overall, I would like to thank all of you for your kind attention. And I would like to always to acknowledge and to thank my collaborator, Thomas Ryan. We did it together, all this project. I mean, this was all conceived together. Chanel Lovett is our technician for histology. Shruti, she helped me for some pharmacological analysis in patch clamp. Diraj with some behavioral experiment at the very beginning. Lily was a technician instrumental for all the injections of the animal, and Susumu, basically my mentor. Thank you very much for your kind attention.
Thank you, Michele, for your excellent talk. And I have a couple of questions. First one is, do you think that there are any local circuits that can regulate the response of these Engram cells, maybe changing this excitability state together with the KIR 2.1 channel?
Look, it's an excellent question. It's not the first time that someone asked me something similar. And I remember in a previous seminar, I had a similar question. And I would say certainly the neuromodulators, they may play a big role. In particular, in the hippocampus, we must take into account noradrenaline. And in part, also the dopamine released by the noradrenergic fibers. That's it. I think this is could be an interesting target. Acetylcholine as well, it could be another target also there. So all these metabotropic receptors, they may regulate this internalization process, for example.
Okay, and do you think that the time between this period of non-recalling and recalling has a kind of hyperconductance state between them, and this basically stop the firing of the neurons or the activation of the neuron?
It could be, it could be. So recording, we didn't do a recording, and anyway, calcium imaging, it won't be extremely instrumental for this kind of things because of the timing of a signal is not great. Electrophysiology will be much better in order to understand. But it's possible that the cell is immediately after recall, it finds itself in a high conductance state, and therefore it must downregulate some intrinsic conductances because of some reverberation at the synaptic level, for example. So it's a possibility, yes, for sure.
Okay, and do you think that there are other kind of channels that are activated in the background of the resting membrane potential that can regulate also this kind of behavior, or it's appear just is geared to one?
So I would say that a second target that could be interesting is the persistent sodium current. This, I think it could be nice to investigate properly. It's not as strong, but is heavily present in the granule cells. And so this can generally is a game changer. For CA1 pyramidal cells, the BK currents have been attributed to the change in excitability due to conditioning. But I suspect IH may play a great role there because they are hyper diffused there. They are everywhere in the dendritic arborization of the pyramidal cells.
Okay, thank you. I think that there is no more question, and I would like to thank you again for your excellent talk, and thank you.
Thank you very much. Thanks a lot.
Okay. Now we have the next presentation is from Dr. Pablo Fuentealba. He's an associate professor at the School of Medicine in the Catholic University in Santiago. Dr. Fuentealba research is focused in the it's focused in the memory encoding and the relationship between different brain areas, such as prefrontal cortex, insular cortex, and hippocampus. He also had very interesting works focused on the role of interneurons in the regulation of gamma oscillations and contextual discrimination. And today, Pablo will talk about the hippocampal contribution to spatial and episodic memory formation. So we would like to thank Pablo to accept our invitation, and you can start when you want.
Thank you, Elias. Can you hear me well?
Yes, yes.
Yes? Okay, so just a second, I'll share my presentation. So can you see that?
Yes.
Okay. So thank you for the invitation, and thanks for attending. So I will just show you briefly the results that we have from two recent projects that we have been working on in the past few years. So one has to do with the connectivity between hippocampus and medial prefrontal cortex. So the way we approached this initially was from the side of stress. So we know that stress is a very important regulator in learning and memory. There are tons of studies on that, particularly in acute stress and chronic stress, right? But in recent years, there has been a lot of attention paid to prenatal stress. That's the stress that the mother experiences while it's pregnant. We know today that this kind of stress is a very important risk factor for neuropsychiatric disorders in the offspring, particularly when they reach adulthood. So we wanted to study in animal models, just very naively, we wanted to find out what were the behavioral consequences of prenatal stress in the offspring. So for that, what we did, we used these two. Can you see my pointer? I'll use this. Maybe this is better.
Yes, we can see it. Yeah.
So we use this very simple procedure, restraint stress. That was a small plastic tube where we introduced the pregnant mice. This was done only during the last week of pregnancy. Mice, they have a 3-week period of pregnancy. We introduced the pregnant mice for 1 hour every day at the same time, every day in this kind of plastic tube. So the pregnant mice was unable to move. And this is particularly stressing for mice. And in order to test spatial memory, we used the Barnes maze that was developed in the late 70s by Carl Barnes. This is some kind of, let's say, Morris water maze, but without water, right? It's a dry maze, and it's very good to test the formation of spatial memory. It's much less stressful for mice than what is the Morris water maze. So the design that we had was we would let the mice have their offspring, and then we tested the offspring when they reach early adulthood, when they were like 2 months, actually when they were 60 days. We started experiments. So first we controlled that actually the mothers were stressed. These are the curves of weight change during pregnancy for control mice and for mice that were stressed during the last week of pregnancy. So the rate of weight gain or gain of weight, it's a marker of stress. You can see that during the last 2 days, there was a significant difference between the pregnant mice, those that were stressed, and compared to the control ones. So this is a suggestion that mice that were stressed during pregnancy, they themselves were stressed, right? But that was not the case for the offspring. So the offspring, we tested them when they were, 60 days old, 2 months, before starting the experiments that I will show. We control their weight, and they gain weight in a similar way to the control mice. So this suggests that the offspring of prenatally stressed, the offspring of mice that were stressed during pregnancy didn't produce stressed offspring. Was that more or less clear? I hope so. We also weighted the suprarenal glands that, usually in the case of stress, they are overgrown, there is atrophy in the glands, and they were not different in weight as compared to controlled mice. So we had the idea that, we had some evidence suggesting that the offspring themselves, before starting experiments, were not stressed. And finally, we tested them in this, the elevated plus maze, which is a standard test to assess for anxiety. And prenatally stressed mice that you can see here, and control mice, they were not different in any measured in the elevated plus maze. So we conclude from this that prenatally stressed mice during early adulthood, they were not showing signs of stress or of anxiety themselves, all right? So having said that, and having done that, we tested the mice in the Barnes maze. So the Barnes maze, basically you put the mouse in the middle of the maze, and you allow the mouse to explore the environment. This is an aversive test for mice because this is an elevated platform. We painted it white. It was a bright white, and there is a strong light shining on top of the maze. So this is the environment is also very exposed. So this is something that rodents really don't like. So they try to hide. They look for a place to hide. And one of these holes has actually a small black box where the mice can hide. So you see that at the beginning, of course, they have no idea that such a place exists. So they just roam around, they explore the maze. But as time passes by, they start finding and learning the location of this escape box, all right? So this is what we actually compute. We compute the latency for the mice to find this place where they can hide. So you see at the beginning, they have no idea that there is a escape box, but towards the end, so we do 4 trials per day. So after 4 days, the animal already, they pick up very quickly the location of they remember the location of the escape box, and they head directly to that place, okay? And this process, the learning process, I mean, the curve of latency to reach the target is pretty much similar between prenatally stressed mice and control mice. So, you know, just from this approach, we cannot say that there is a difference during learning, all right? But after learning, what we do, we test memory in this very same environment. The way to test memory, well, there are several ways, but the way we used or we approached to this issue was on the 5th day after training concluded. On the 5th day, we exposed again the animal to the environment, but we blocked the access to the escape box. So we do that on day 5, and this is a test of short-term memory because they just finished training the day before, or we do it at day 15 in a different group of animals. And if you do it 10 days after finishing training, that is what is called long-term memory, right? So what we found, if you see here, is that latency to find the target between both groups for short-term memory or for recent memory was not different, okay? So the day 5, after finishing training, prenatally stressed mice and control mice, they remember the location of the escape box similarly. So the surprise came for the remote test. So what we found was that actually the ones who remember very well the location of the escape box were the prenatally stressed mice, not the control mice. Actually, control mice, they pretty much forget about the location of the escape box. So our interpretation of this was that this kind of phenomenon could be approached, could be assimilated as some kind of post-traumatic stress disorder for mice, because they exhibit the persistence of a memory that was aversive for them. All right. So having found this behavioral phenomenon, then we switch to identifying neurophysiological correlates of this phenomenon. So for this, what we did was we anesthetized the animals that performed the test immediately after performing the recent memory test or the remote memory test, and we recorded simultaneously in the hippocampus and the medial prefrontal cortex. So we recorded the dorsal hippocampus, CA1 area, and in the prefrontal cortex, we focused on the infralimbic and prelimbic areas. So we recorded with glass electrodes. We used pipettes because we wanted to record single units, and we wanted also to label some of the cells because initially we wanted to do some neuroanatomical analysis of labeled neurons. So very briefly, what we have here, this is the firing pattern of a single unit, control mice, recent memory, remote memory, and prenatally stressed mice and recent memory and remote memory. So you can see here that prenatally stressed mice in the prefrontal cortex, this is in the prefrontal cortex, we found actually a hypofunctionality of this cortical region, in the sense that neurons were discharging significantly less than in prenatally stressed mice as compared to control mice. And not only that, but you know, the number of units that we were able to identify during the penetration of the pipette in the cortex was also lower. So there was a hypoactivation and generalized hypoactivation of the prefrontal cortex, specifically for the case of prenatally stressed mice. So, of course, we don't have evidence for what I'm about to say, but this somehow suggests that, you know, if the prefrontal cortex is the one that controls extinction in the amygdala, right? So probably a group of those cells projecting to the amygdala are hypoactivated. So you don't get the activation of the intercalated cells in the amygdala that are the ones that control extinction, particularly fear extinction, right? This is just speculation, but it's kind of consistent with the data we got. So we labeled some of the cells, like I told you. We did some kind of coarse visual analysis. We didn't see any aberrant alterations like you see in, for example, chronic stress, and histologically, the tissue also looked pretty well preserved. So this seems to be some kind of, I don't want to say subtle, but I don't have a better term now. But this is not like a gross alteration of the nerve tissue like it happens particularly in chronic stress, right? So we also compare, we measure, I'm not showing here because of lack of time, but we focus a lot on cortical oscillations, and we didn't see any major changes due to prenatal stress. Remember, this is anesthesia. This is just the first approach. We compare theta, delta oscillations, low waves, ripples, whatever you want. We didn't see any changes. But where we found an interesting result was in the connectivity, using as a proxy the hippocampal ripples, you know, sharp wave ripples, these fast frequency periods of activity. We heard about them yesterday. Using this as a reference, because this was shown actually very nicely during the late 90s by Matt Wilson and his group. Sharp wave ripples can drive cortical activity in the prefrontal cortex also. They can drive spiking. So what we did was to use this ripple as a reference to compare it with the spike timing of the prefrontal cortex. And this is where we found a difference. Again, this was a subtle difference. In the case of animals that were tested for the recent memory, for the formation of a recent spatial memory, there was no difference in the cross correlation between ripples and spikes in the prefrontal cortex. But the spiking in the prefrontal cortex was kind of depressed for the case of prenatally stressed mice in the remote memory condition, right? So basically, we found hypoactivity or of the prefrontal cortex at the level of unit spiking, which was not accompanied by like gross, coarse morphological modifications. And we also found a subtle but still statistically significant difference in the spike timing, in the functional connectivity between prefrontal cortex and hippocampus. So we see that these results, in a preliminary way, they suggest that the coordination between hippocampus and cortex is important for memory formation, and this is altered, this is disrupted in pathological conditions such as the one we just modeled with prenatal stress. So in the next part, what we did was to approach this this phenomenon, but now we wanted to study this process, the learning, the acquisition of the task. Actually, we wanted to study memory, but we found something in between, which is what I'm going to show you now. So this we did with chronically implanted mice. So we developed this this kind of drive, and we inserted 3 tetrodes in the prefrontal cortex and 1 tetrode in the hippocampus to use it as reference. And after animals recovered from surgery, they learn the Barnes maze. You see that learning is something that takes place normally, despite the fact that mice have like a second head on top of them. They can still learn they can still acquire the task pretty efficiently. And we noticed in the previous study that I showed you that you have this kind of phenomenon, which I also show you. At the beginning, animals don't know where the escape is. But at some point, they realize that there is escape, and they start looking for it. But as training progresses, I show you that latency decreases, and they manage to go directly to the escape box. And the way they can do that is because they use spatial cues that are, located in the room where they are tested, because the environment itself, the platform, is pretty homogeneous. So the idea here is that this has been described particularly from other fields, in people studying behavior in rodents, ethological behavior, that there are different strategies of navigating an environment. So the idea here is that when you learn where the location is, you have to use the environmental cues to be able to navigate to your target environment. So obviously the difference in latency to reach the escape is quite large in this case. So we focus on these 2 types of navigation. One, which is called non-spatial, basically, when the animal still doesn't know very well where the escape box is, and the one that is present in the last trials, which is spatial type of navigation strategy. Names are not really original, but we didn't invent them, so we're just using what the literature does. So we had these simultaneous recordings, I told you, of prefrontal cortex and hippocampus during the task performance. And we did something very simple. We just calculated the synchrony, the broad synchrony between these 2 signals. We just computed the coherence, the wideband coherence between these 2 signals as animals, progressed in the learning the task. And what we found was that this band here, the gamma band, particularly the slow gamma band, increased as animals, acquire the task. So as they move from the non-spatial strategy to the spatial strategy, the synchrony in gamma between hippocampus and cortex increased, okay? This didn't happen for theta oscillation, the theta oscillation, which is also very important in the coordination, first in spatial navigation for mice, right? And it's also very important to coordinate hippocampus with surrounding cortical areas, also with prefrontal cortex. This is something that, Matt Wilson and Chiapas, they showed also in the 90s. Not the same result, I mean, the coordination between theta in the cortex and the hippocampus. So we found this specific increase in the coordination between hippocampus and cortex during acquisition of the task. But not only that, also we found that the prefrontal units, neurons in the prefrontal cortex that couple with these oscillations, theta oscillations and gamma oscillations, they also exhibited a stronger phase coupling with the background oscillation, with the theta oscillation or the gamma oscillation. The coupling was stronger during the period of navigation. So this is something I didn't mention, sorry. We had three epochs in the test. So first we put the mouse covered with a box in the center of the field. So the mouse doesn't know where it is because it's covered, right? So we leave it there for 1 minute. Then we lift up the box, and the mouse can look for the escape. And then once it reaches the escape, we keep recording for one more minute. So we have these three phases of the task. One is the start when the mouse is inside the box. Then during navigation, which is very variable, right? Because it can take a long time for them to reach the escape, or it can be very quickly when they use the spatial strategy. And then we also record when they are in the escape box. So what we found was that phase locking between prefrontal units and the background oscillation, whether it was theta or gamma, phase coupling was stronger when the animals implemented the spatial navigation strategy as compared to the other kind of strategies, right? And even more, I know this is not, this is not linear, but this is just an approximation. This is a way of showing that there was a correlation between that coupling, the coupling between the prefrontal units with theta or gamma oscillations. So stronger the coupling, the more efficient was the navigation, in the sense that the latency to find escape was shorter, right? And the second result I want to show you in that sense about units is that there was this phenomenon of ramping up in the relative firing rate of units as they approach the goal, only for the case where animals implemented the spatial navigation. That means, towards the last period of training, when they went directly to the escape box. This had nothing to do, I'm not showing it here, but this is something we also computed. We calculated if this had to do something with theta, with the frequency of theta oscillations, and it was not. So the frequency and power of theta oscillations was not really different between the spatial strategies and non-spatial strategies. So this was kind of specific. So, there was this stronger phase coupling of prefrontal units to the theta and gamma oscillations during spatial navigation when they implemented the spatial strategy, and there was this ramping up of activity towards the goal that then disappeared once the animal entered. So this is interesting because this is, I think it's interesting because it's analogous to this ramping up of evidence accumulation that you see in decision-making tasks. Probably it has nothing to do with that, but I think it's an interesting observation. And the same as with the phase coupling, this correlated with behavior. So the stronger the firing rate, the shorter the latency of the task. So what I'm trying to say with this, again, is another way of saying that the functional connectivity between hippocampus, in this case, dorsal hippocampus, remember, this is dorsal CA1, which is not the hippocampal region providing the main input to the prefrontal cortex. The connectivity between hippocampus and cortex seems to be relevant for spatial memory formation. And the second part, this will be brief. Yeah, this was a different study, but it also had to do with the role of connectivity and the importance of hippocampus, but now in the formation of contextual memories. So you know that we just heard in the previous talk that dentate gyrus is important, among many other things, for the role that it has been attributed to it, which is pattern separation. So pattern separation is supposed to help to discriminate overlapping representations, environmental representations, and overlapping memories, right? So again, we had a very simple approach to this. We wanted to study how this contextual separation of overlapping representation takes place in the dentate gyrus. So there are a lot of studies here, of course, including what we just saw in the previous talk. What we wanted to see here was the role of interneurons. So in the cortex, in the hippocampus as well, we have these two main types of inhibition. Actually, in the next talk, we are going to hear a lot more about that. We have the parvalbumin expressing interneurons that typically innervate the perisomatic region of principal cells, right? In the cornus ammoni regions, they target the soma, proximal dendrites of pyramidal cells. In the case of the dentate gyrus, they target the soma and proximal dendrites of granule cells, right? But then we have these somatostatin cells that provide inhibition to distal dendrites. So we actually wanted to study the role of these somatostatin cells because they target the region where the input from the entorhinal cortex reaches the dentate gyrus. We saw a very nice, quite a few nice pictures in the previous talk showing the innervation from the entorhinal cortex to the distal dendrites of granule cells in the dentate gyrus, and this is massive. This is the main input to the dentate gyrus, right? The perforant path. And these cells, somatostatin cells, they selectively target the dendrites of granule cells in the same place where entorhinal input reaches. So we were wondering what would be the role of these neurons in controlling the activity of the dentate gyrus during conditions of pattern separation or, discrimination of overlapping contextual information, which is more accurate for our conditions. So for this, we got a couple of mice strains from Jackson laboratories. So one of them expressed CRE in somatostatin cells, and the other strain expressed the halorodopsin and YFP. So we crossed these animals, we genotyped them, and we tested a few of them just to see if they were expressing YFP. So we wanted to know if this YFP was actually somatostatin, right? So we did some immunos. This what I'm showing here is from another study. We used the same strains in that study. We focus here on the basal forebrain. We could not do it for the dentate gyrus because this was, towards the end of 2019, which was kind of a shaky period in this country. So we actually could not complete the histological experiments, but I show you the evidence for the basal forebrain. So we stained for choline acetyltransferase. This is to identify whether the cells were expressing the machinery capable of producing acetylcholine. So here you see a YFP neuron. Here you have another one, and they don't co-express chat, right? They didn't express parvalbumin either, and most of them, they co-express somatostatin. So that was kind of a relief. And then we tested, this was a collaboration with Andres Chavez in Valparaiso. We tested whether those neurons expressing YFP that were somatostatin positive were expressing functional hyaluronidopsin. So he got some of these mice and he tested, he performed, patch clamp recordings, and he shone light on them, and, cells that were expressing somatostatin, YFP that were positive for somatostatin. They also had the functional opsin, in this case, hyaluronidopsin inhibitory. It was not the case for cells that were not expressing YFP. So this was quantified, and it was kind of a reliable result. And then we tested now we move to the dentate gyrus. We tested in vivo the effect of silencing these neurons in anesthetized mice. So here we show the track of the electrode that was inserted in these mice. And you see when we shine light, we have a mixture. This is kind of normal. You have cells that decrease the firing rate, others increase, and others don't change their firing rate. So by definition, the cells that were inhibited were our somatostatin cells. So we did some cluster analysis. We were trying to identify, just based on the certain features of spiking activity, we were trying to see if we could separate into other types of interneurons like parvalbumin. In this case, all is putative, right? Because we don't have controls for this, but it's kind of, it fits the picture. And the broad spiking cells were probably mossy and granule cells. Anyway, we wanted to be sure that this was working before performing the behavioral tests. So we did two kinds of tests. The first one was a spatial memory test. This was a collaboration as well. In this, we collaborated with Pedro Beginstein and Noelia Weisklop in
Aló.
Creo que tenemos un problema con el internet, tal como dijo ayer Pancho, un Zoom sin problemas de conexión, no un Zoom. Le vamos a avisar a Pablo. Un momento.
Okay. My back?
Yes, you're back. Okay.
Funny. Okay, so where did you lose me?
A few seconds ago, maybe 10, 15 seconds ago.
Okay, so I was showing this. Did you get this?
No, the before, I think.
Okay, so I start from the beginning, all right?
Yeah, okay.
Here, okay.
Yeah.
My God, bugger. Just a second. So can you see my screen? Yes?
Yes. we can.
Okay, so I was explaining this. This is the last part already. So I was explaining the first test. So I was telling you that the mouse has to learn to discriminate these two objects from this object placed here in the last part of the test. So that seems very easy, right? But it's not that easy for mice when you start bringing the objects closer and closer together. So at some point, the mouse doesn't distinguish or doesn't discriminate, is not able to separate this object from these two objects, all right? So this is what we show here. So we measure the discrimination index. A positive discrimination index tells you that the animal explores more the novel object than the two called familiar objects. So when the animal sees this object that's different than these two, it spends more time exploring this object, all right? But if you as you bring together the objects, at some point, which is this one, when they are very close together, the objects, the animal doesn't discriminate this object from the other one, and it explores both, both conditions, these two objects compared to this one, it explores both objects, or both conditions in the same amount. And then you have a discrimination index of 0. So it explores both of them at in the same amount, proportional. So important, control that exploration times, exploratory patterns are not affected in this kind of test. So what we did here, we took our transgenic animals and we shone light during the exploration, during the encoding of the first condition. So here we took the intermediate separation, because why we did that? Because for pattern separation to work, representations have to be overlapping. So if the objects are very far apart, the theory tells us that probably the dentate gyrus will not be recruited. And if the objects are very close together and the animal doesn't discriminate them, then there is no pattern separation, right? So we need a condition, an intermediate condition where we are pushing the system, but it's working in the window where it's designed to work. All right. So during exploration of this environment, what we saw during optogenetic inhibition of somatostatin cells, what we saw that the discrimination was compromised. So these are control animals. In this case, these are transgenic animals, but not expressing functional halorhodopsin. But when the animals were expressing, sorry, I would put the pointer. Yeah, so this is the case where transgenic animals were expressing functional halorhodopsin. So we basically abolished the capacity of the animal to discriminate these two conditions. When somatostatin cells are not inhibited, the animal still can perform the discrimination. So this was what we did for a spatial memory test. We also developed a contextual memory test. So in this case, the animal has to explore two different environments, okay? And these environments are kind of similar. They are overlapping. So you have two, this is what is called the object in context. So you have two objects, one particular context, and then the animal explores two different objects in another different environment, but these two environments are kind of similar. And then in the testing condition, what we do is we take one of the objects from the other environment. So this is a mismatch object, and when the animal identifies this object as novel, as misplaced in the context, it explores more the novel object or the mismatch object in the novel environment. Sorry, in the known environment. All right. So here we compute again the same exploration index, and we repeat the same type of test, but now we have a more dramatic differences between contexts. So in this case, the environment is squared, and in this other case, the environment is triangular, okay? So this is a more radical difference. So we call this one the similar condition and this one the dissimilar condition. So first we tested whether the dentate gyrus was actually necessary, was recruited for this kind of test. So for that, what we did was to infuse MBQX. We block AMPA receptors during encoding and also during recall. So when we block AMPA receptors during recall, there was no effect. I'm not showing that. But we did find an effect when we were blocking encoding. So if you infused, in our case, when we infused MBQX, when we block AMPA receptors before encoding of both environments, then animals were not able to discriminate these conditions as compared to this one, okay? The dissimilar condition is analogous to what I was showing you here. The dissimilar is a condition where objects are really far apart. So the dentate gyrus is not recruited. The function of pattern separation is not recruited because the representations are not overlapping, all right? So this is why in the dissimilar condition, even though we block AMPA receptors, there was no impact in performance. Probably because, this is our interpretation, because the pattern separation function of the dentate gyrus is not recruited. But it does have a significant effect when representations are overlapping. That's in the similar condition. And then we tested our hypothesis that somatostatin cells are relevant for this. So we showed that the dentate gyrus is necessary to perform this test. That is some kind of pattern separation test. Actually, I mean, it's object in context, overlapping context test. So what we did now was to repeat the same experiment, but in the transgenic animals. So when we shine light during recall, there was no effect on performance, but we found it during encoding of both both environments in the similar condition. So this is pretty much the same what we found with the drug. So basically, you need functional somatostatin cells in the dentate gyrus to separate overlapping representations of the environment. When they are not overlapping, then you don't require somatostatin cells, and probably you don't require pattern separation, the pattern separation function of the dentate gyrus. So basically, what I have shown you are two pieces of evidence that support this idea that the hippocampus is required for the formation of contextual memories and spatial memories, and the connectivity of the hippocampus is actually very relevant for those functions. So I would like to acknowledge Francisco Aboitis. He's a long collaborator from a long time. He was a collaborator in the first project I show you, and in the second one, well, the first project was developed basically by Ignacio Negron. He's currently in the Universidad de Valparaíso with the help of Nelson Espinosa. The second part I show you is basically the thesis project of Christian Morales, who finished last year. And like I told you, this was a collaboration with a group in Argentina. So thank you for your time, for your attention, and I will be happy to have some questions, comments, or discuss some of this data.
Thank you, Pablo, for your excellent talk. I have a couple of questions. First, the first one is during the prenatal stress, do you explore if there is some kind of critical time when you can give this stress during pregnancy that can affect or have more effect in the connectivity between the basically the
right. Yeah, the short answer is that we didn't. I just can tell you something about the design is that since we were going to, I mean, we wanted to focus on the connectivity between hippocampus and prefrontal cortex. And prefrontal cortex, you know, it develops, is one of the last cortical areas that develops during gestation, during pregnancy. So this is why we stress specifically the mothers during the last week. We didn't want to develop to stress the mothers during the entire pregnancy, because that would be maybe far too unspecific, and probably it would constitute some kind of chronic stress procedure for the females. So we focus on the last week of pregnancy, because we knew that the prefrontal cortex develops during that last week. And particularly interesting was the fact that this we learned afterwards, that it seems that dopaminergic innervation of the prefrontal cortex also develops during the last days of pregnancy. But we didn't play around with that, you know. It's not like yeah. So
I understand. And do you have any idea of if there is some kind of a structural change in the neurons in the prefrontal cortex after the prenatal stress in the mothers, in the mice, basically in the offspring? Sorry.
Yeah. So this is something I didn't do myself, but Francisco, he has an article on that. He studied that, and they found some changes in methylation of few genes. So there is some kind of structural modification, but like I showed in the talk, actually there was no like gross or coarse. We didn't find like big changes in the tissue or in the neuronal morphology. So this was kind of something affecting connectivity, probably.
Okay. So and the other question is, in your experiment with somatostatin interneurons, do you have an idea of what kind of local circuit can be involved in this effect, in the effect that you are observing in the connectivity?
Yeah. So our idea is very simple, you know. Actually, during the time that, so this was the idea, and actually it was corroborated. Because when Christian was doing the experiments, which you know how this takes it's a half an hour presentation, but it's 3 years of work. So the thing is that our idea was very simple. That somatostatin cells, we thought that they were regulating the size of the engram, that they were controlling, because you have all this massive volley that arrives from the entorhinal cortex that depolarizes the granule cells, right? So granule cells are known to you know, you have more granule cells than pyramidal cells in the rest of the hippocampus, but they have very low levels of activity, spontaneous activity. So the idea is that they are constantly inhibited by these parvalbumin cells that control the spike timing, and whatever the other cell type is there in the dentin gyrus. So what we thought was that the entorhinal input depolarizes the granule cell, but it also feed-forward activates somatostatin cells. It's more complicated than that, because the somatostatin cell is in the hilus and is activated by the MOSI cell. But whatever, let's say that it's activated when the input from the entorhinal cortex arrives. So our idea was that if we remove that input, if we remove that cell, the somatostatin cell, from the circuit, there will be less inhibition that controls the size of the population of granule cells activating. So we will somehow disrupt the differential representation of input from the entorhinal cortex. I don't know if that's clear what I'm saying.
I understand.
Yeah, the idea is that we will have more granule cells activated that should not be activated because they are not being inhibited by somatostatin cells. And while actually, Christian was doing the experiments, there was a nice article that showed exactly that, but for fear conditioning.
Okay.
Probably, actually, it was probably someone from Tonegawa's lab. I don't know.
Okay.
Maybe, yeah.
Okay. In honor of time, Paulo, we would like to thank you for your presentation, and we will follow with a break between 5 to 10 minutes, and we will be back with the presentation of Dr. Chai-Yu.
Yeah, thank you.
Thank you.
Thanks to you.
Profesor Elías.
Aquí está.
Sí, haremos la prueba con la próxima panelista que nos pidió que pudiéramos antes de, en el break, hacer la prueba.
Ok.
Hello.
Hey, Chai-Yu. Hello, Chai-Yu.
Hi, how are you?
Good, and you?
Good. So how should we do this? Should I just click share screen?
Yes. Yes. Share screen, and you told me that you have video, so we can test if the video works well.
Okay. So can you see my screen right now?
Yes. Yes.
Okay.
Can you put it in the presentation mode, please?
Okay.
Yeah. Perfect. Yeah.
All right. So let me just go to a. It should work. It works on my computer, and since I'm using the same.
Okay.
All right, so let's just.
It is perfect.
So yeah. And that's really, there's another one, but it should, if that one works, this should work too.
Yeah.
Okay, great. Just yeah, looks, everything is working. So
yeah, perfect.
So I should unshare now, right?
Yeah, but we will take a 5-minute break for the people just to see, because I forgot to mention before that after there was a break, so I went directly through Paula. So
yeah, so people need to stretch their legs. Okay,
yeah.
So I will come back like 5 minutes or something like that.
Yeah.
Okay.
Yeah.
All right. See you later.
Yes.
Perfecto.
Hello again. Now we will start with the presentation of Dr. Chae-Yu Chu. Dr. Chu is an associate professor at the Interdisciplinary Center for Neuroscience at Valparaiso, Interdisciplinary Center for Neuroscience of Valparaiso, and at the Faculty of Science at the University of Valparaiso, and also she is the CIMB Max Planck Tandem Research Group leader. Her research focuses on the balance of excitation and inhibition and how glutamatergic synapses that mediate excitation modify their strength and number during learning and memory. Her group uses several advanced optical and pharmacological and genetics tools to dissect the Gabaergic circuits and type of interneurons, modulating the different response of the in the neurons. Okay, so today Dr. Chu will talk about the diverse mechanisms of Gabaergic regulation on neuronal dendrites. So thanks, Chae-Yu, for accepting our invitation, and please, you can take the stage whenever you want.
Okay, so thank you, Elias, for the opportunity to talk a little bit about some of the work we're doing. And let me just check. So everyone can see my screen?
Yeah, we can.
Okay, so let me just go to pointer. Okay, so we've so far heard amazing talks today and yesterday about how neurons, and in particular neuronal firing in groups and populations work together to mediate some forms of learning and memory. And what I like now for the next 30 minutes or so is to kind of switch gears a little bit, okay? And concentrate on the neuronal dendrites, right? So this is the region that's receiving these synaptic inputs that we've been talking about, right? And so in the cortex, these inputs are chemical. So they're either mediated by the release of glutamate or by GABA, right? And so in the case of glutamate, this is excitatory. You get these depolarizing excitatory postsynaptic potentials that bring the cell closer to firing. And that's why they're called excitatory. And GABA is inhibitory because it actually triggers the hyperpolarization of the membrane, which brings the cell further from firing and therefore is inhibitory, right? And we've already heard, as Pablo alluded to before, that these cells, there are different types of cells that mediate excitation and inhibition, right? So in the cortex, this is depicted in red, a pyramidal cell, which is excitatory, releases glutamate. And in green are Gabaergic cells and are inhibitory, right? And so the question has always been, how do excitation and inhibition on synaptic inputs in neurons, how do they contribute to shape the final firing pattern of these cells that we've been talking about? And this issue is complicated because if you actually blow up the dendrite, you'll see these ultrastructural images of spines that protrude from the dendrite with the terminals of these excitatory cells impinging on these spines, right? So this is the actual excitatory synapse that we talk about, right? And in the case of inhibition, we also see similar things, right? So it's a little more diverse because you have Gabaergic synapses that form directly on dendrites, and that's thought to be a major way these inhibitions work. But you also have inhibitory synapses on the spines themselves, which also are receiving these excitatory inputs, right? So what's the effect of these Gabaergic synapses compared to these on shaping excitatory function and overall neuronal firing, right? So this is the question that we want to address. And we've heard so far a lot about what excitation is doing, and that's because it's generally thought that excitatory cells are the projecting cells, right? So they are conveying information from cell to cell, from brain region to brain region. But we do know some things about what Gabaergic inhibition does, right? So it does shape the way neurons fire and how much they fire and what patterns they fire. It shapes the synaptic plasticity of excitatory signaling, and that is thought to underlie the learning and memory that we've been talking about. And they also shape brain plasticity during development that Pablo alluded to a little earlier, as well as shaping these rhythmic oscillatory activities that we also heard about mostly yesterday for hippocampus. And we do know that inhibition is very important because when you have disruptions in Gabaergic inhibition, you have disorders, right? Epilepsy, schizophrenia, autism. But the question is, how do these cells work, right? And this question is really complicated by the fact that GABA actually activates GABA A receptors and also GABA B receptors. So these are ionic receptors, and they're formed by different compositions of these 5 subunits, right? So there are many different types of alpha and beta and these delta subunits. So, and they shape the way these GABA receptors will function. And I won't talk about this as much, but GABA also triggers G protein coupled signaling, intracellular signaling, and they're coupled to different partners in the cell that will shape its signaling, its impact on the cell also, right? And as we heard a little about, the Gabaergic cells are diverse, right? So you can identify them by different proteins that they express. So pyrobamin is one, somatostatin we heard a little bit about, and there are these ionotropic serotonin receptor subtypes, right? And so for generally speaking, pyrobamin makes up about 30% of Gabaergic cells in the cortex. And they typically have a special targeting pattern, right, that we saw in the hippocampus, which is close to where the cells, the soma, and where the cells actually initiate action potentials, right? And the somatostatin and serotonergic cell types are more dendrite targeting, right? So they target where the synaptic inputs would be to these neurons. And actually, most of the Gabaergic synapses are on dendrites. And there's about 58% that's directly on the dendritic shaft, plus the 26% that's perhaps on the spines. And that varies a little bit depending on the region that you're talking about. Right. And so why is it important to look at neuronal dendrites? Right. And that's because the dendrites themselves, they don't just passively carry information from the synapses to the soma that then drive firing, right? So here's a schematic taken from a review by London and Hauser a little while back that shows the different kinds of things that neurons actually do, right? So there's the normal passive propagation of signals from synapses to the soma, and that typically will attenuate, right? It'll get smaller and slower, right? That's a passive process. But dendrites also have active properties, right? And so here is an example where synapses on the same dendritic branch will actually synergistically potentiate its impact on the cell, right? Compared to synapses that are across different dendritic branches, right? So there's a rule of synapses working better together as a dendritic branch than across, right? And here's an example of a coincidence detection, right? So dendrites are active. So if you actually fire an action potential from the axon initial segment, the action potential back propagates down the dendrite. And if it coincides with activation of a particular set of synapses, you can actually trigger these supralinear calcium signals, right? That can then have a role in changing the way the dendrite is computing its signal, right? And here is what is called a logical operation. So what happens is an and or logic, right? So if certain synapses are active, they will inhibit the impact of other synapses, right? And so here's an example of an excitatory to an inhibitory putative function, right? So generally speaking, dendritic inhibition is thought to suppress excitatory signaling, right? And this I will talk a little more about, right? First, I just want to say a word about a special property of dendrites, right? So if you were able to record from the soma and from the dendrite and stimulate in the synaptic region, what you can trigger are these calcium signals, right? These in the dendrite, and they can act superlinearly, right? So that can potentially be signaling in the dendrite. And sometimes they backpropagate, they propagate into the soma and therefore trigger action potential, but not always, right? So here's an example of when they can act independently, right? So you don't have to actually ultimately end up with a firing of the cell, right? Even though you now enhance the dendritic excitability of the cell. And this kind of calcium signal, the supralinear summation of calcium, is important for the synaptic integration, right? Because synapses are thousands of synapses, right, on dendritic, on neuronal dendrites. And so how do these synapses work together, right? And this calcium can shape plasticity and can shape learning. So in vivo models, people have shown that these calcium spikes is critical and is required for learning, right, in certain tasks. And so what is Gabaergic inhibition doing? We do know some things, right? So Gabaergic inhibition on dendrites can reduce the backpropagation of these action potentials, right? And they've also been shown to even block these dendritic calcium spikes, right? And they can block NMDA receptor signaling through some signaling cascade that requires protein kinase A. But how is it doing this? Right, and so the question, as I alluded to, is not a simple one because you have different cell types that mediate GABA transmission, right? And they do target different places of the cell, right? And the receptors themselves are diverse, right? And they actually are differentially expressed along the dendrites themselves. So today I will talk briefly about two studies. One is a slightly older one, so I will go rather briefly and quickly with this. And a second study that shows two different ways that Gabaergic inhibition can change the dendritic signaling of neurons, right? And so the first study, as I alluded to, we were interested in understanding what somatostatin interneurons do because they are a primate. They make up about 30%, sometimes maybe even 40%, depending on the cortical region you're talking about, of the number of Gabaergic cells. Right? And these are critical because they are actually the major dendrite targeting Gabaergic cell in the cortex. And so what we wanted to know is how these interneurons modulate neuronal dendritic signal. And so to do this study, we're combining the epigenetic activation that you heard about today, where we put these channel rhodopsin channels into some interneurons specifically, right? And we wait 2 weeks, and then we make slices from them, and we can now activate these some interneurons specifically with light, with this blue light channel rhodopsin. And this is what you see in the prefrontal cortex. And so the second technique that we need is to be able to image activity in the neuronal dendrites themselves, right? And here we take advantage of 2-photon fluorescence microscopy, right? And so here's an image of what a typical layer 2-3 pyramidal cell looks like in the prefrontal cortex. And then what we do is we patch these cells and we load them with 2 dots, right? So there's a red dye that just lets us see the dendrites and the spines, and then a calcium indicator, in this case flow 5, so we can monitor calcium, right? And calcium is important because it's a proxy for neuronal activity, but calcium in itself is also important because it's a mediator for many forms of synaptic plasticity that is important in learning and memory. Right, and so here, I'm just going to show, wait. Sorry. Right, so here is a movie where we inject through the soma a depolarizing current, right? That'll trigger action potential in the soma, and that back propagates out into the dendrites, and you'll see this as this green signal, right? That's pretty global. That's stretching along the dendrites and the spines. Okay, I need to stop this. Okay, stop. All right. And so in order to monitor this calcium activity fast, right, what we do is instead of making a movie, we line scan. We perform scan over line just repeatedly, very fast, over 2 milliseconds, right? And what we can do then is we can see the calcium signals in our region of interest. So here, for example, is the spine and the dendrite, right? And we're just performing line scans repeatedly, right? So now we can monitor in time. And what you see here is the increase in the green signal, which is calcium, as a result of injection of action potentials. And we can see that this calcium signal is mediated by all the different types of voltage-gated calcium channels, right? And you can see this by the pharmacological blockers for the specific calcium types. Okay, and so what is happening to the calcium signals by activation of some interneurons? And so to do this, what we did is we injected action potentials into the soma, and we paired it with optogenetic activation of the some interneurons, right? And then we just compare what is the signal in the calcium with just the action potential alone, or with co-activation of the interneuron with the action potential. And so indeed, what we can find is that the inhibition mediated by the somatostatin interneurons does reduce calcium signals in the dendrites and the spines, right? And what we found was that this inhibition required the participation of high voltage calcium channels, right? The blockade of low voltage activated channels did not seem to have an effect on the ability of somatostatin interneurons to inhibit calcium signals. Okay, but what was more surprising to us is when we actually looked at the ability of the inhibition to modulate calcium signals in nearby spines, what we found was that the inhibition can be very local, right? In this example, only spine 1 showed inhibition of calcium signals, whereas its neighbors did not, right? And so as a population, we saw that only the spine of reference showed calcium inhibition, and generally that inhibition goes away as you go further out from that spot. Right? And here's just a linear regression to show that there's no relationship. Okay. And so that was a BAP propagating calcium signal. What about its effect on glutamatergic signaling, right? And so here what we did, what Gary did, was he took advantage of the ability to focally activate synapses, right? So here, if you shine light in this region, what you can trigger is this local calcium signal, just in the spine. Right, just in the spine that's activated and not anywhere else, right? And we know that this calcium is mediated by NMDA, okay? And so what he did was then used this uncaging of glutamate, right? And first he showed that this inhibition is local as well. This applies for glutamatergic signaling as well as for the BAP-induced calcium signal. And next, what he did was he wanted to see what is happening with synaptic integration, right? So he basically uncaged glutamate, right, in 3 different spines, right? And he will trigger 3 calcium signals in each of them, and we'll get excitatory postsynaptic potentials recordings in the soma, right? And then what he did was just test what happens to this excitatory potential when you have GABA activated, right? And so here in black is what happens when you sequentially activate 3 spines, right? You have this linear building of EPSCs. And when you pair that with GABA activation, you can reduce this summation, right? Which is plotted here. So you have a linear summation, and when you have GABA, you make it sublinear, right? So the synapses are less able to potentiate each other's effect, right? And this is occurring through the NMDA receptors, because if you put the CPP blocker, you inhibit this linear summation, right? And then you occlude the effect of GABA. Okay, and so this is a summary of the first talk. We also did many other ways of looking at this, but at the end, the basic model that we built is one where somatostatin interneurons make their inhibitory synapses directly on spines, right? So the somatostatin interneurons are contributing to that 26% or so spine Gabaergic synapses. And by being able to directly inhibit spine heads, they can reduce calcium, right, in the spine, and they can reduce the calcium signaling that's induced by the glutamate synapse that's on it, but only here, right? It's very focal, only here, and not in this spot, and not in the dendrites. And in addition, this effect can suppress synaptic integration. Okay. And for the second part, so while we were doing the study, and when we wanted to confirm that this inhibition was indeed mediated by GABA A receptors, we found something that was a little confusing at the time, right? Which is that when you block the GABA receptors themselves, you can reduce the calcium, right? So far we showed that GABA inhibition from the somatostatin neurons suppress calcium, right? But here, if you block the GABA A receptors, you also suppress, right? And we don't see this if we just float in a CSF, right? So here's a time course. So what type of inhibition could be doing this, right? And so we first confirmed that you know, if we go to more physiological temperatures, so 32 degrees Celsius, because these experiments were done at 28 degrees, we can still see this inhibitory effect. And then even if we reduce the concentration of the antagonist, here is gabazine, we can still see this inhibitory effect. And even if we switch to a different GABA receptor antagonist, picotaxin here, we can still see this effect. Okay, so what is this? What type of GABA inhibition is mediating this? And so far I've been talking about the synaptic inhibition, right? Where GABA is released and it activates synaptic receptors, right? And this is typically what you see for, which is another word is phasic, right? You can see this fast onset and decay, right? There's another form of inhibition that's termed tonic, right? So this is mediated by GABA receptors that are outside of the synapse, and they're typically formed by different subunits, right? So here, the GABA A receptors of synapses is usually a gamma 2 subunit containing, whereas the extrasynaptic are either alpha 4 or alpha 6. Alpha 5 can be both extrasynaptic and synaptic, right? And then what you see is normally what you, if you block GABA, you will see this tone, right? And so what we wondered is perhaps maybe tonic inhibition is involved here. And so what we tested I'm not really going to show you the data, but we also tested alpha 4 and alpha 6 using a FIP, which is an agonist for this receptor, and we didn't find an effect. So I'm just going to show you what we found for alpha 5. Now, alpha 5 is interesting because it's different. Normally, extrasynaptic GABA receptors have high affinity, right? Because they're further away from the synapse, and therefore they need to be able to detect lower levels of GABA. Alpha 5 are different. They actually have lower GABA affinity, right? Even if they're extrasynaptic. And they've been linked to many disorders, right? So what we then teamed up with Maria Clemencia in Roche, and where she has these negative allosteric modulators and positive allosteric modulators for the alpha 5 containing GABA A subunit receptors. Okay, so what we found was that if you inhibit selectively the activation of alpha 5 subunits, we can also mimic the effect of the general GABA A receptor antagonist, right? You see a reduction. Right? And when we use the positive allosteric modulator, which you can see already here, you actually can increase them. So actually modulating the activity of alpha 5 receptor, GABA A receptor subunit activity can bidirectionally modulate the calcium in the spine. Okay, and so how is this doing this? Because it is tonic inhibition also generally has been shown to reduce calcium signaling, right? The inhibition in general suppresses. So how can this be? How can tonic inhibition now facilitate calcium signaling? And so to understand this, we turn to a model, right? So Tom, what he did is he built a 2-compartment model, right? So he simulated the conductances, right? So the sodium and the potassium conductances and the intrinsic membrane properties of the soma and in the dendrites, right? And then what we basically did is we wanted to see what happens when you generate an action potential in the soma that propagates down in the dendrites. And so the new thing that we added in this model is 2 things. So first, we added a tonic inhibition, right? So what happens when you have tonic inhibition in the dendrites? And the other thing is we added these voltage-gated calcium channels, right? And there are 2 types. There's the low voltage gated activated and the high voltage activated. And so what you see when you initiate action potential in the soma is when you block or not block, there's really minimal effect in the soma, right? But what you found was that when you block the GABA conductance, you end up having this slight depolarization of the dendritic membrane. And that led to a slightly faster action potential, right? But now if you looked at the calcium current, right, and also the conductance from these voltage-gated calcium channels, you see that when you block GABA conductance, you actually reduce calcium current, right? And this is mostly due to a reduction in the low voltage activated calcium channels. There's a little bit of the high voltage, but it's mostly in the low voltage. And how is that happening? So we looked at the activation gate and the inactivation gate, right? So that's the special thing about the low voltage gated calcium channels, is that they fast activate and fast inactivate. And when you block GABA, what you find is this lower change in the opening, right, of the low voltage calcium channels. And they are less of them that's going to be open by the inactivation gate, right? So the 2 of them together actually end up leading to this reduce in calcium conductance. And so the next thing we looked at is how does this effect change with dendritic distance from the soma, right? And so we found a surprising effect where what tonic GABA is doing is leading to this slightly hyperpolarizing membrane potential as you go further out from the soma. Okay, and when you block the tonic GABA conductance, you prevent this gradient, right? So it kind of depolarizes, but the effect is bigger further out, right? And then, so what happens is that the effect by blocking GABA conductance then also reduces the calcium as a function of distance from the soma. And then black is an example of the effect of blocking GABA receptors if there was a mix of low voltage gated and high voltage gated calcium channels. But if you only have high voltage activated, the effect is much smaller, is very, very minimal, right? Whereas if you have only low voltage activated calcium channels, you can get a very potentiated effect, right? So this model has two predictions. First is that the inhibition by gabazine of the calcium signal is going to be stronger as you go further out from the soma. And two is that this effect should require more the low voltage activated channels compared to the high voltage. Right, and so experimentally we looked at this, right? So if we just measure calcium signals and the effect of gabazine, we see that gabazine can inhibit the calcium signals more the further out you go. Right? And so what about the low voltage activated channels? So low voltage activated is the T-type, right? And so the nice thing is that there's a selective blocker for T-type. So first, what we wanted to show is, can T-type channels really be de-inactivated by hyperpolarization? Right? So here's an example of the calcium signal that's obtained when the cell is resting at -60 millivolts, right? Which is what our recordings have been. And so T-type has some modest contribution to the calcium signals. When you hyperpolarize the cell, you can see that the calcium signal increases relative to -60, right? So if you compare black here and here, there's more of it. And the contribution of the T-type is bigger, right? So when you block T, the calcium signals go back to what it would look like at -60, right? So most of this increase in calcium at -85 is due to T-type, right, being de-inactivated. So what happens to the Gambazine effect? So if you block T-type channels, right, then the Gambazine has a much smaller effect compared to controls, right? It's diminished. But if you block the high voltage activated calcium channels with a cocktail, right? So here's minomolapine and conotoxin. Blocking high voltage activated calcium channels does not prevent gabazine from reducing the calcium signals, right? So it seems like tonic GABA is acting through low voltage gated calcium channels. And so what is the effect? What is the effect of this calcium modulation by tonic inhibition? And so here we took advantage of this process called depolarization-induced suppression of inhibition. Right? So this is a protocol where if you increase calcium signals in the cell, right, either through an actual potential burst or through just depolarizing step, you trigger calcium that will end up producing endocannabinoids. So what endocannabinoids do in this case is 2-AG. There are many different types of endocannabinoids. And so in this case, what 2-AG does is it travels back to the presynaptic site and activates these specific receptors, cannabinoid type 1 receptors in the brain. And the activation of these receptors ends up suppressing the release of GABA in this case, right? And so what you end up seeing is this transient reduction in GABA release, right? And that recovers after 30 seconds, right? And so here you can see in black is the iPSC before, and then in gray is the iPSC immediately after this depolarization. And so what we did is then we applied this low dose of gabazine, right? That does inhibit calcium right? That's induced by this train. But this low-dose gabazine doesn't block all GABA transmission, right? So you can still get an iPSC, right? But what you find is that the action potentials now doesn't suppress inhibition. Right? So it looks like tonic inhibition is facilitating the calcium signaling that then can facilitate synaptic plasticity, in this case, short-term synaptic plasticity. And if you recall, we showed that when you modulate the alpha-5 GABA-A receptors, right, you can bidirectionally suppress or enhance calcium signals. So do these receptors also have a role in DSI? And yeah, indeed. So in the Gabazine case, right? So you have cells that are depolarized, but when you apply Gabazine to block the GABA receptors, you see that the suppression is less. Right? It's closer to 100% baseline. And NAM mimics this effect of Gabazine, right? You can see that the DSI is reduced, right? And in the case of the positive allosteric modulator, it can enhance DSI, right? So in this case, the 50 Hertz action potentials cause a bigger suppression of inhibition. Okay. And so now the summary, basically what we found taking these two studies together, right, is that there are two forms of inhibition. There's one that is synaptic, and it's on spines. And the way it works is very localized, right? It inhibits the calcium signals in the spines, right? And that can have a role in synaptic integration, very synapse-specific kind of things. On the other hand, you have these tonic GABA-A receptor activation, right? And what it does is it enhances the release of, in this case, endocannabinoids that then can go back and activate CB1 receptors on Gabaergic terminals that then can suppress inhibition. Right? It has this weird role where inhibition can reduce further inhibition, right? Tonic inhibition modulates synaptic inhibition, right? And if you block these receptors, you reduce the calcium signals, reduce the ability of the neuronal neurons to inhibit synaptic inhibition, right? And so it's actually interesting to think that maybe there are other physiological roles for this. So this is an example of a study in the human cortex where they showed that you can have activation, glutamatergic activation, either depicted in path X or pathway Y. And the activation of this glutamatergic synapses causes these dendritic calcium spikes, right, that we talked about, right? And they each will trigger these dendritic calcium spikes if you record from the dendrite. But when you co-activate pathway X and pathway Y, you actually suppress this dendritic calcium action potential. Right, so it has this anti-correlated coincidence role. And what's interesting is if you add inhibition to this, you can recover the ability of these inputs to drive dendritic action potentials. Right, it's not been shown, but it would be enticing to think that maybe this tonic inhibition might play a role in facilitating calcium signals in this way. Right. And another open question is, of course, where these GABA signals are coming from, right? And it is possible that these GABA receptors are just tonically active on their own. Another possibility is that perhaps these are being released by interneurons that are tonically active, or they like to burst, right? And this remains to be investigated in the future. And with that, I'd like to end, and thank you for your attention.
Thank you, Chai, for your excellent talk. And I have a couple of questions. First of all, do you know where precisely is located this type T-type calcium channels? Do you know the pattern of expression? If it's postsynaptic, or in the dendritic shaft, or any
Yeah,
implications?
so very good question. So we don't know. Actually, this is ongoing project in the lab, right? So what Camila is doing is basically triggering action potentials and then measuring calcium signals in different regions of the dendrite. Distal dendrite, distal apical dendrite, somatic apical dendrite, and basal. And then basically applying calcium blockers and seeing if there's some pattern. And right now we don't know yet, but there's definitely some interesting patterns of calcium signaling that is regulated by the membrane potential. So if you recall, when you hyperpolarize the cell and then do a BAP, right, the calcium signals increase. And that increase actually has a somatic dendritic pattern, right? But what we haven't done is tested to see whether that change in somatic dendritic calcium is mediated by the T-type or not. So that still remains to be determined.
And do you think that there is a kind of a local circuit that is basically inhibiting the interconnecting interneurons before to go to the pyramidal neurons and show or explain this effect, like I don't know,
for the tonic
bag or
inhibition?
fit? Yes, yeah.
Yeah, so what I didn't say is, so what I manipulated was the GABA transporter in neurons, the GAT1. And that didn't do anything on the calcium signals. Of course, that, there's the astrocyte GABA transporter, right? That might be, and I didn't test that. So first, it remains to be seen whether modulating GABA concentrations by changing transporter function or something like that can change tonic inhibition. So far, at least for the neuronal type, I couldn't do it. So I don't know whether even GABA release from which cells are important, astrocytes or neurons. But the fact that the neuronal transporters didn't do something kind of leads me to think that maybe it's not the interneurons, but maybe the astrocytes.
Okay, got it. And the last question that I have is, do you have this kind of back propagated action potential? So you know if in this layer 2-3 pyramidal neurons, you have another channels participating, like HCN or something like that, or even potassium channels, or any conductance activated by calcium, basically, that can be in the close neighborhood of this T-type calcium channels.
Yeah, so that's another good question. So we haven't tested it ourselves. There have been suggestions that there are these Ka channels and also the IH channels that do play a role. They're differentially expressed in neuronal dendrites. It's not quite clear if these particular layer 2-3 pyramidal cells in prefrontal cortex do it or not. What I can tell you is, so normally these channels have an effect in attenuating calcium propagation along the dendrites, right? So if you looked at CA1 pyramidal cells, or if you looked at layer 5 pyramidal cells in cortex, you see this decrease of calcium signal as you go further out in the dendrites. And that is because of the activity of the IH and the Ka. In layer 2-3, I did not see such drop. So I image calcium in spines that are 200 microns away. You really can't go that much further for layer 2-3 pyramidal cells. And there doesn't seem to be this decrease as you go further away from the neurons. So that either suggesting that these conductances don't play a role here. They don't have that differential expression higher in distal dendrites, or there's some kind of compensatory mechanism in the layer 2-3 pyramidal cells that compensates for these conductances. I don't know.
Okay. I understand. And one question that just is, do you know, maybe it's not your focus right now, but you have any idea of if any of the interneurons in the layer 1 are doing the same in the pyramidal neurons you can see in this in your results, basically somatostatin.
Yeah, so you mean like the Cajal-Retzius cells?
Yeah.
So I don't know. I haven't looked at that specifically. It's very possible. It's very possible, but that these cells also, because they do burst, they do mediate also distal inhibition. I just haven't looked.
Okay. Thank you, Chay, for your excellent presentation, and thank you for accepting our invitation. And thank you.
Thank you.
Okay.
Thank you.
We have another talk from Dr. Errol Miller from MIT. It will be start at 1 p.m. He's an expert. I think it's the... Sorry, voy a hablar en español. Tenemos la siguiente presentación a la 1 de la tarde del Dr. Errol Miller. Él es un experto en corteza prefrontal y memoria de trabajo, y los invito a todos a que vengan. Él es el experto a nivel mundial de esta área, así que los esperamos en 20 minutos más. Por eso, nos vemos.
Unos segundos, profesor, denos unos segundos que estamos haciendo unos arreglos con la introducción, por favor. Solo 2 minutos antes de que volvamos al panel.
Oh, now. Yes. Hi, Aaron. How you doing?
Good. How are you?
Yeah, not too bad. Not too bad.
Excellent.
Yeah, things are going well so far.
Good to hear.
Yeah. Yep.
We are about to start in about 2 minutes. Errol.
Okay.
Yeah, I see
Are
they're
solving some problems with the translation, so because you will be translated to Spanish.
Okay.
Yeah.
We have 2 channels of audio, so one for you and one for the translator.
Should I share my screen now?
You
For
can
testing, yeah, that'll be.
yeah, you can test your presentation.
Listo, profesor, todo ok.
Okay, thank you.
How's that? Good?
Yeah,
Yep.
it looks like it's covering more. It's higher than the screen. I think.
Sorry?
The screen is oversized, so we cannot see the whole characters in your presentation.
I
Oh,
think
is that right?
I saw them all.
Oh, yeah, I think it's fine. Yeah.
I think it's your problem, Elias. Not
It's my problem. Okay.
Yeah, I can see everything. Yeah.
Sí, se ve bien. Se ve bien.
Okay. So it's my problem. Sorry. I don't know what I did.
I won't worry
Okay.
about it then.
Yeah. I guess, yeah. Okay. Yeah, it's me. Okay. So we will gonna start. I will. Okay. So thank you everybody to be here today. I will present Dr. Earl Miller. He's a professor of neuroscience at the Pickauer Institute for Learning and Memory and the Department of Brain Cognitive Science at MIT. Dr. Miller uses experimental and theoretical approaches to study the neural basis of high-level cognitive function that underlie complex goal-directed behavior. He focused on the frontal lobe, the most elaborated region of the human brain that is linked to neuropsychiatric disorders. Errol is an expert in working memory. I don't have any enough words to describe how important is his work in this field. So today he will talk about something else about working memory. So thanks, Errol, for accepting our invitation, and please, you can start talking when you want.
Thank you very much for the kind invitation. I really appreciate the chance to speak to you. And yes, I will be talking today about working memory. We all know what working memory is, perhaps especially this group. Working memory is the ability to hold things in mind. It's the foundational mechanism that allows higher level cognition, because with working memory, an important thing to remember about working memory is that it's not just a short-term memory buffer, it's under volitional control. And that's the most important thing about working memory, because working memory allows us to take in information and wait to make a response, maybe take in more information, decide to respond later, hold off decisions. It allows us, working memory is the fundamental mechanism that allows us to not just react to the environment, but to wait and act when we want to act on the environment. So it's very critical to high-level cognition. So the question today is, how do we hold thoughts in working memory? Now, the classic model for the past 50 years or so has been persistent spiking of neurons. The idea here is that when you're holding something in working memory, whether it's a visual image or it's a set of numbers you have to type into a keypad. You're doing it by having neurons in your cortex that are constantly firing, keeping those memories alive, keeping them in working memory. And that's illustrated here from a classic figure from Pat Goldman-Rakish, one of the pioneers of the neural basis of working memory. And what it shows here is a typical neuron that has been used to support this persistent spiking hypothesis. This is one neuron. It's averaged across multiple behavioral trials. The neuron spiking rate is on the y-axis. Time is on the x-axis. Here's where you present the monkey, in this case, it's monkeys, with a visual cue that they have to hold in working memory. Then there's a memory delay, and then the monkey makes its response. So what typically happens is you present the animal with a cue, you get elevated spiking, reflecting that cue. Once the cue goes away, spiking drops down a little bit, but it's maintained at a rate above baseline rate. And this presumably is keeping the ensemble, the memory, the information about that cue online and available in working memory over this memory delay until the animal makes its response, then activity drops back down to baseline. Now, this has been the dominant model for the past 50 years or so, and it's mainly based on averaging across, in this case, one neuron across multiple trials, or multiple neurons across multiple trials. However, over the past 20, 25 years or so, there's been great advances in multiple electrode technology that allows us to record not from one neuron at a time, but multiple neurons at a time, up to hundreds of neurons at a time. And this allows us to have a more detailed examination of what is happening in real time when animals are holding information in working memory. And what's especially important here is what's happening in real time on single trials. Neurophysiologists average across multiple trials because they want to increase the signal-noise ratio. The idea is that averaging brings out the signal and all the noise cancels out. But what if some of that noise is actually signal that we're just averaging out? And so our lab and a number of labs have been looking at neural activity in real time on single trials. And what this has revealed is that in actuality, most neurons in prefrontal cortex and other higher cortical areas, prefrontal cortex being the frontal lobes, it's the part of the brain that's most important for most central working memory. Not the only part of the brain, but it's a central part of the brain. And what this has revealed is that most neurons in places like the prefrontal cortex, parietal cortex, higher level cortical areas that contribute to working memory, most of them don't show real robust, persistent, constant firing over this memory delay. Instead, they show very sparse and bursty firing. And I'll show you an example of that in a moment. But this observation that if you look really carefully at a finer grain detail, that most neurons do show this kind of sparseness, not persistent activity. It looks persistent if you average a bunch of bursty neurons together or average them together across multiple trials. But this observation that it is on single trials in real time that this activity is sparse and bursty has led to several updates to the classic model. One update came directly from Pat Goldman-Rakish, the pioneer of working memory, of the neural basis of working memory, who in one of her last papers showed that it's not just spiking that seems to carry working memories, it's spiking plus spiking-induced temporary changes in synaptic weights. And she showed this in biology of the prefrontal cortex. When neurons in the prefrontal cortex spike, they spike, and then for about 750 or milliseconds or a second later, there's a temporary potentiation of the synaptic weights between connected neurons. And that, Goldman-Rakish and Wong hypothesized, may help carry working memories in between these episodes of spiking. There's also the activity silent model of Mark Stokes, who makes the same sort of argument, and a more recent model called the dynamic attractor model of Michael Lundquist. And I'll tell you about that dynamic attractor model in a moment. But all these updates to the classic models say the same thing. They say that spiking is not doing all the work. Spiking is important. Elevated levels of spiking during this memory delay is indeed important for holding information in working memory, but the spiking is helped along by temporary changes in synaptic weights, temporary impressions that spiking leaves in the networks. And this makes sense because spikes cost energy. Every time a spike occurs, it's costing a lot of energy. So rather than have constant spiking carrying the working memories, there's occasional spiking, sparse spiking, and in between the sparse spiking, the changes in synaptic weights are help carrying those working memories. And that saves energy because, again, spikes cost energy. But it also makes working memories more robust, less easy to disrupt than if they were held by persistent activity alone. Persistent activity in working memory is modeled by something called attractor dynamics. And attractor dynamics simply model this persistent state of activity where the neurons are constantly firing. And one thing we've learned from all these models is that attractor dynamics, persistent spiking, is not a very robust way, not a very good way to hold information in working memory. Because if a distractor comes along or a new input that comes along, you want to add something to working memory, the new input disrupts the attractor dynamics, disrupts the persistent activity, and changes the memories. So models of working memory based on persistent activity are very easy to disrupt. And these activity silent dynamics are more robust because the synaptic weight changes are more resistant to interference. So here's what we did to test this model. I'll tell you about the dynamic attractor model of Michael Lundqvist. Michael came to my lab to test this model. And the way we tested this model is we studied a task in which monkeys had to hold two visual items in working memory simultaneously, while we recorded from multiple electrodes in the lateral prefrontal cortex of the monkey, shown here. Here's the prefrontal cortex. Lateral part of the prefrontal cortex would be right here on my head. So this is anterior, that's posterior, that's dorsal, that's ventral. I assume you can all see my cursor. Yes? If not, please let me know.
Yes.
Great. And here's the task. The task is the monkey was shown in the encoding phase, two samples, two colored squares at two different locations. Then there was a memory delay, and they were tested for the memory of those two samples by a test array, and the monkey had to make a same and different judgment. Were these two squares the same as the ones they saw earlier? Now, this design looks complex, but the important thing to remember is all that was required of the monkey was to hold two visual items, two colored squares in the location of working memory. That's it. Just hold these two items simultaneously in working memory. And here's what we found. This shows, now not showing spiking activity here, what I'm showing is power from the local field potentials. Local field potentials are the mass average of millions of neurons spiking together in a coordinated fashion. We looked at the spiking activity too, but what we found is illustrated best by looking at the local field potentials. So here's frequency on the y-axis. This is frequency of the local field potential, anywhere from 0 hertz up to 120 hertz. The power of the local field potential is shown in color. So red is more power and blue is less power. Time is on the x-axis. S1 and S2 is when the two squares are shown to the monkey. Here we're showing the monkey the first square. There's a short pause. The second square, the monkey has to hold these two squares in working memory over this memory delay and then make a response out here. And what we found is illustrated in this slide. We found, like other studies, that there was spiking activity that conveyed information about the two pictures the monkey was holding in working memory, the two items in working memory. But that spiking activity that carried the working memory was associated with bursts of gamma from the local field potentials. So whenever the spiking occurred that conveyed information about the two items, S1 and S2, there was also co-occurring these bursts of gamma. And you can see that here, these gamma bursts and the spiking was most evident when the monkey was encoding the picture in working memory during the time in which we showed the picture to the animals, and then periodically over the memory delay. We saw these sparse bursts of gamma over the memory delay, and those bursts of gamma were associated with the spiking that was carrying information about working memory. So the first observation is that the spikes carrying the working memories were associated with gamma bursting in the prefrontal cortex. Now, another thing you may notice that here's the spiking up here on the top of the slide in the gamma range from 40 hertz and above. There was also bursts of a lower frequency in the beta range from about 30 hertz to about 20 hertz. And that's what's shown here. Here's two examples of beta bursting in this lower range. And what we found is that the beta bursting was also there, but it was anti-correlated with gamma. So you see the gamma and the spiking that carries the working memories, that encodes the information in working memory and carries the working memory, is associated with these bursts of gamma. And there were bursts of beta, but the beta tend to occur whenever gamma wasn't occurring. So here we see two bursts of beta here, and they're occurring at this interval of time where there's no gamma. Okay, so beta and gamma are anti-correlated. And this is illustrated here, where now we're looking at not the individual trial, but the average across multiple trials. We're looking at the burst rate. How often do we see these gamma bursts shown in the orange color versus the beta bursts shown in the brown color? And you can see they're completely anti-correlated with one another. Gamma bursting goes up when the monkey is viewing the two sample objects here during S1 and S2. The monkey is seeing S1 and S2, and there's a burst of gamma. The monkey sees S2, there's a burst of gamma. And whenever gamma bursting is going up, beta bursting goes down. And over the memory delay, what you typically see in the prefrontal cortex in spiking is you see there's a relatively low level of spiking early in the delay that builds up over the course of the delay as the animal gets ready to use the working memories. And you see that reflected here in gamma. The gamma bursting associated with the spiking starts out at a relatively low level, then builds up over the memory delay, reaching an apex near the end of the delay when the monkey has to then use the working memories. And we see a corresponding drop in beta bursting. So beta and gamma are like mirror images of one another. Whenever gamma goes up, that's the gamma that's carrying the spiking, that's carrying the working memories. Whenever gamma goes up, beta goes down. Whenever beta goes up, gamma goes down. So I've said repeatedly that gamma is carrying the spiking that's carrying the working memories. If gamma and spiking is associated with the contents of working memory, what does beta do? Well, the answer is that a number of studies in our laboratory have shown that beta rhythms, these lower frequency beta rhythms from about 12 to 30 hertz, carries top-down information, the animal's knowledge about how to perform the task. And this is first shown by Tim Bushman, who's now a professor at Princeton. He was a graduate student and a postdoc in my lab. He had the animal switch back and forth between two different rules. The animal was looking at the stimuli, had to make judgments about either the color of the stimulus or the shape of the stimulus. And we cued the animal on this trial, pay attention to color, ignore shape. Then on another trial, pay attention to shape and ignore color. So the monkey is following these two rules, color or shape. And what Tim found is that this is recordings from the lateral prefrontal cortex. Here's the arcuate sulcus. There's the principal sulcus. Posterior is here, anterior is here, dorsal, ventral. All these little circles are a different recording site where we had a single electrode. We had an array of electrodes. And what we're looking at here is, again, local field potentials, but we're looking at which recording sites showed an increase in synchrony whenever the animal was following the color rule versus the shape, or what we'll call the orientation rule. It wasn't shape, it was the orientation of the item, but essentially the same thing. And what we found is that when we look at these patterns of LFP synchrony across the prefrontal cortex, we see different patterns of synchrony for one rule versus the other rule. Okay, so different shifting patterns of beta synchrony. And we looked across all frequencies to look for this effect, and we only saw it in the beta range, 12 to 30 hertz. but there's different patterns of beta depending on which rule the monkey was following, as if these patterns of synchrony are forming the ensembles needed to solve this task, needed for the animal to follow these rules. Okay, is there a question? No. Okay, so once we saw this effect for these rules, we then applied it to other forms of top-down knowledge. We saw the same thing for spatial categories, taught the monkeys the categories up versus down, and same things, different patterns of beta coherence in the prefrontal cortex and the parietal cortex, and between the prefrontal and parietal cortex, when the monkey was making a judgment about one category up versus the other category down. We saw the same thing for shape categories. We had a study where we taught animals to recognize the categories cat versus dogs, and the same thing, different patterns of beta coherence across the prefrontal cortex whenever the monkey was judging cat versus judging dog. And finally, other examples of shape categories, this was formed by dot patterns. The take-home message from this slide is that whenever we teach the monkey some rules, some knowledge that it needs to solve the task, not simply, the animal's, it's not a matter of the animal viewing a picture. Whenever the animal is using some acquired knowledge, some top-down knowledge to solve the task, we see this top-down knowledge reflected in patterns of coherence in the beta range, in the prefrontal cortex, and in other cortical areas. So what this suggests to us is that we have this push-pull relationship between beta and gamma. Gamma is carrying the contents of working memory. It's involved in encoding information in working memory and maintaining information in working memory, and it's anti-correlated with beta. So the idea here is that top-down information is carried by beta, and they play an inhibitory role that controls the gamma that encodes information in working memory and maintains information in working memory. So it's almost like a beta is this control signal that turns on and off the faucet of working memory storage. Okay? So beta has this inhibitory relationship to gamma. If you want to encode information in working memory, you drop down beta. That allows gamma to be expressed. That encodes information in working memory. You want to maintain information in working memory, you keep beta at a low level. And if you want to drive up the contents of working memory, like we see over this working memory delay, so the animal can get ready, when the animal's getting ready to use the working memories, we drive down beta, it allows more expression of gamma. Okay, so it's a beta acts as a control signal, inhibitory control signal that turns on and off working memory storage. That's the basic idea. And please, if you have any questions, not being clear about anything, feel free to interrupt me. Okay, so once we have this observation, we want to explore this further. This anti-correlation between beta and gamma suggested, when we showed the anti-correlation between gamma and beta, we showed that gamma carries working memory, beta carries top-down information, but do they really work like this push-pull control signal that actually controls working memory? So we did this in a more recent study where we did a very complicated working memory task. I'm not going to walk you through, in the limited time we have here, I'm not going to walk you through this particular study. I'm going to highlight the results. I'm going to invite you to look at read the paper if you want more details. But what we found is that evidence that gamma and beta and gamma work together to gate information in working memory. So first of all, we showed that beta gates information into working memory. Whenever the animal wants to encode information in working memory, beta drops. It allows gamma and spiking to increase. And you see that right here. Here's the animal has to encode the stimulus in working memory. Beta drops. It allows gamma and spiking to start encoding the information in working memory. So that can be seen here. Well, we also showed in this study in 2018 in Nature Communications that beta is also involved in clearing out working memory. If at some point in the trial, the monkey is cued to know that the working memory is no longer relevant, the monkey can just forget the working memory. What happens is there's a big increase in beta that drives gamma down, spiking stops, and working memory is cleared out. Okay? And another thing in this particular study we did is we had a trick where we had the animal switch the contents of working memory. First, the monkey was holding one stimulus of working memory. Then we gave the animal a cue, forget about that stimulus, and code a second stimulus of working memory. And when that happened, there was this rapid cycling of beta gamma. That first beta went up and dropped down the spiking in gamma to the first object. That cleared the first object out of working memory. Then beta dropped, gamma increased, and the second item was then encoded in working memory. So this study in 2018 shows that you do see this push-pull, this dance between beta and gamma that is very suggestive of a control signal that controls the encoding and maintenance of information in working memory. Right? And importantly, what neurophysiologists have used for many years to see whether the spiking effects or any effects they're seeing are actually related to the task at hand, in this case, related to the working memory task, we look at error trials. If the animal makes an error, can we see the error reflected in whatever activity we're looking at? And that was true for this beta-gamma balance. In fact, we looked at individual trials and tried to predict what mistakes the monkey would make, what decision the monkey was making. We found that the balance between beta and gamma was a better predictor of the monkey's decisions and whatever errors the monkey would make than spiking activity alone. So beta and gamma was highly predictive of what decisions the monkey would make, even when the decision was wrong. Okay, so we wanted to look more closely at the dynamics between beta and gamma, and we did so by doing so-called multilaminar recordings across using multiple contact electrodes along the layers of cortex. And this is relevant because the superficial layers of cortex are the feed-forward layers that carry sensory information from the back of the brain to the front of the brain. Whereas the deep layers of cortex are the feedback layers that carry top-down information from the front of the brain to the back of the brain. And to make a long story short, what we found is that these dynamics were reflected in different layers of cortex. So the spiking activity that was carrying the working memories was mainly in the superficial layers of cortex. Here's the different cortical layers. The dotted line shows the middle layer, the input layer, layer 4. The color in this case shows spiking activity. Here's a working memory delay where the monkey's seeing a cue here. This is the memory delay. And we see there's spiking that's carrying the working memory the monkey's holding, in the stimulus the monkey's holding in working memory, but that's mainly in the superficial layers of cortex, not the deep layers of cortex. And that makes sense because the monkey's holding a sensory stimulus, a working memory, so that should come, since it's feed-forward sensory information, it should be in the superficial layers, and it is. And then we looked across the different layers. Again, here's the different layers again, with 0 being the dotted line being the middle layer. We saw more gamma in the superficial layers and more beta in the deep layers. And this makes sense because, again, gamma is associated with the sensory information the monkey's holding in working memory. Superficial layers, the feed-forward layers. Since gamma is associated with holding sensory information in working memory, it should be stronger in the superficial layers, and it is. And beta is carrying top-down information, we hypothesize. And because the deep layers are the feedback layers of cortex that carry top-down information, it makes sense that beta should be stronger in the deep layers of cortex because it's carrying top-down information feeding back from the front of the brain to the back of the brain. And the same beta-gamma anti-correlation I showed you earlier, we saw taking place across the superficial and deep layers of cortex. To save time, I won't walk you through this slide, but what this figure shows is that whenever beta in the deep layers of cortex was strong, gamma in the superficial layers is weak. And whenever gamma in the superficial layers was strong, beta in the deep layers was weak. So again, this push-pull relationship between beta and gamma, but happening between the two cortical layers. So what this suggests is the following. Top-down information is carried by deep layer beta from the frontal lobe back to sensory cortex. Deep layer beta regulates weaker superficial layer of beta. We see beta in superficial layers too, but it's a weak compared to deep layers. So we think that the superficial layer beta is entrained by deep layer beta, and the superficial layer beta in turn inhibits, gates or controls the gamma spiking carrying working memory stored, thereby turning on and off working memory. Okay, so that's the basic model. Now we have this top-down beta information acting as a control signal that gates the expression of gamma and spiking that encodes information in working memory and maintains information in working memory. We decided to carry this further in a very recent experiment just published by looking at a curious thing about visual working memory. Now, you probably all know that vision is split between the right and left brain. The two halves of both retinas go to different sides of the brain. The right half of each retina goes to the left brain, and the left half of each retina goes to the right brain. Bottom line being that your right brain sees one side of vision, one side of gaze, and your left brain sees the other side of gaze. Now, it's long been thought that this split between vision is well known for visual processing back in posterior cortex, and certainly in area V1 and primary sensory cortex. And there's been a sort of a tacit assumption in the field that somehow this split, left and right, gets united at the level of higher level cognition and prefrontal cortex. But we found out back in 2011, again, work done by Tim Bushman, Simon Kornblith, and Demetrios Panissis in the lab, they showed that the split between right and left is even true at the level of the prefrontal cortex and in working memory. In fact, your right and left sides of vision have separate working memory capacities, as if the monkeys, and we've replicated this in humans, as if monkeys and humans, as if we are split brain patients, two separate brains, one looking at the left side of vision, one looking at the right side of vision. So that's weird. We have right and left is split between the right brain and the left brain. But if that's the case, how do you actually unite the left and right sides of vision? When I'm looking around, when I'm playing Frisbee, when I'm just operating in my daily life, I don't notice the split between right and left. So how does the brain do it? So we trained monkeys on this paradigm where we had them encode information in one side of vision, then did a little behavioral trick that encouraged them to switch the working memory from one side of the brain to the other side of the brain. So here's what we did. We had the animal fixate its gaze on one side of the computer screen, and we presented a stimulus the monkey had to hold a working memory to the one side of that gaze. In this case, this monkey is looking right here. We're presenting a banana to be held to working memory on the right side of vision. That means it's going to the left brain and not the right brain. Then on one half of the trials, we played a little trick on the monkey. On half of the trials, when the monkey was not, the banana is already gone, the monkey's just holding the banana in working memory. And while the monkey was holding the banana in working memory, on half of the trials, we had the animal move its gaze to the other side of the computer screen. Now, if the banana was still on the computer screen, when the monkey moved its eye to the other side of the screen, that should switch the vision from one side of the brain to the other. But in this case, the monkey is not looking at the banana, the monkey is just thinking about the banana. What happens there? Does this move the stimulus from the one side of the brain to the other? Well, the answer is that it does. So here's the two possibilities here. What we have here is neural activity. The contralateral stimuli are stronger than the ipsilateral stimuli. That's true all over visual cortex. It's also true for motor cortex. Your left side of your brain controls the right side of your body, so contralateral stuff is a stronger representation. One possibility is shown here is the dark line, the dark green line, the dark brown line are what happens on the trials in which there is no saccade and the memory just stays in the original hemisphere in which it was encoded. So in this case, that'd be so you should see a stronger response to contralateral stimuli, a weak response to ipsilateral stimuli. And what the lighter colored line shows is what happens when we have the animal make the saccade and the memory switches from the contralateral hemisphere to the ipsilateral hemisphere, or in the case of light brown, from the ipsilateral hemisphere to the contralateral hemisphere. Now, one possibility, this little trick does nothing. The memory just stays where it is. And we expect to just see no change whatsoever. But the other possibility is that the memory switches. So in this case, if the memory switches from contralateral to ipsilateral, we should see this flip where the stimulus started out with a strong representation because it was on the contralateral side. Then the monkey makes its saccade, shown here in the this line right here, and that just flipped the memory to the ipsilateral side, and then its response should weaken. Correspondingly, if the stimulus started on the ipsilateral side, shown here by this light brown line, and then the saccade moves the memory to the contralateral side, it should suddenly get stronger after the saccade because the memory is now switched to the contralateral hemisphere. And what we found is this: the memory didn't switch to the other hemisphere. So that's shown here. Here's the trials in which the here's the beginning of the delay. You see the stronger neural responses to the contralateral stimuli, to the versus ipsilateral stimuli. This is before the saccade, in the middle of the delay. And this is the trials in which the saccade occurred. And first there's a burst of activity in the saccade, then the memory switches from the contralateral hemisphere to the ipsilateral hemisphere. And indeed, we see this dropping down of information, of neural activity. Once the saccade is made, the memory switches, neural activity drives down to the same level that you'd expect if the stimulus was on the ipsilateral side to begin with and never moved. And correspondingly, if a stimulus starts out on the ipsilateral side, then the saccade occurs. There's an increase in activity to that stimulus to the same level that you'd expect if the stimulus just started out in the contralateral hemisphere. So you see the switching operation. Contralateral stimuli after the saccade switch to the ipsilateral hemisphere, and ipsilateral stimuli after the saccade switch to the contralateral hemisphere. So the memory traces, once the saccade is made, the memory traces disappear from the ascending original hemisphere and then reappear in the opposite hemisphere, the receiver hemisphere. And interestingly, the same dynamics we saw for beta-gamma dynamics we saw for working memory gating are also true for transfer of working memories between the hemispheres. Here's around the time the memory is being transferred, and around the time of that memory transfer, there's a decrease in beta power and an increase in gamma and theta power. Gamma and theta tend to co-track in cortex. Gamma and theta, they work together. So we see this decrease in beta and increase in gamma during the working memory transfer, just like we saw for working memory maintenance. If you want the memories to transfer from one hemisphere to the other, you drop beta down, and that allows the gamma to then transfer and be expressed in the other side of the brain. And a Granger's causality analysis revealed that the directionality of the synchrony was consistent with memory trace transfer. So here's a member of the Granger causality here on the y-axis. This is for the sender to receiver hemisphere. The memories are going in the direction that we'd expect, versus the opposite, receiver to sender. The memories are going in the opposite direction, not the direction we'd expect. And here's the frequency of the LFE synchrony on the x-axis. And as you can see, the Granger's causality measure is stronger in the sender to receiver direction than the receiver to sender direction. It's stronger in the direction as if these synchrony is playing a role in pushing the memories from one side of the brain to the other. Okay? And the last thing I'll tell you about is the most surprising thing that we found, is that we found that when these memories transfer from one side of the brain to the other, it actually changes the ensemble, changes the pattern of activity that represents the memory. So what we did is we trained a classifier on the pattern of activity across an array of electrodes in the prefrontal cortex. And we trained the classifier on the trials in which the stimulus started out in the contralateral hemisphere and then just stayed in the contralateral hemisphere. It didn't go anywhere, just stayed in the contralateral hemisphere. Then once we trained the classifier on those trials, we then applied it to working memories that were in the contralateral hemisphere, but were transferred over from the ipsilateral hemisphere. Okay, so we're training the classifier on memories that started on one side of the brain and stayed there. And then we're testing that classifier on memories that were received from the other hemisphere. And the hypothesis is that you get the same pattern of activity. You simply copy the working memory ensemble from one side of the brain to the other, and it doesn't change. You're simply copying it like you're taking a picture of it and just pushing it over to the other side of the brain. But the null hypothesis is that there is no transfer, that the working memory actually changes when it's transferred. And that's exactly what we found. So here's the trials in which we trained the classifier on the working memories that stayed in the contralateral hemisphere, and then tested them on models on working memories that started in the ipsilateral hemisphere, but then transferred to the contralateral hemisphere. And there was virtually no transfer at all. And what this says to us is that, so the classifier trained on visually invoked activity in the contralateral hemisphere cannot be used to decode the exact same working memory, same object, same location, same everything, but it can't be used to decode that working memory if that working memory was transferred from the other hemisphere. So it seems as if when the working memory is transferred from one hemisphere to another, its pattern of activity, its ensemble, completely changes to a new format during this transfer. So I see I'm running short on time, so I'll skip the last experiment just to say that we found that the same beta gamma dynamics not only has a role in working memory control, it also plays a role in something called predictive coding. And I encourage you to look up this paper in PNAS published last year, showing the same beta gamma dynamics play a role in how the brain regulates processing of sensory information using predictive coding. But to sum up, since I'm out of time, I'll say that we believe that these studies show that alpha beta is like a control signal that feeds top-down information in the deep layers of cortex from the front of the brain to the back of the brain. These top-down alpha beta signals, and by the way, I'm using alpha beta here because when you look at the back of the beta is primarily in the frontal cortex. When you move to back most posteriorly, there's a down regulation of frequency, so the beta in the frontal cortex becomes alpha in the visual cortex, but it's essentially the same signal. So these alpha beta control signals carrying top-down information regulate the expression of gamma that carries the spiking that carries bottom-up information, the contents of working memory or the contents of perception. And we believe this is a modal cortical circuit that plays a role in top-down control of basically everything: working memory, attention, predictive coding, and even the transfer of working memories between the hemispheres. We have looked now, our original studies were in the frontal cortex and focused on working memory. We have looked for these dynamics all over cortex, and we're finding it everywhere we look. Every time there's a bottom-up sensory information, more gamma. Every time you need to regulate that sensory information, there's an increase in beta or alpha, depending on where you are. And that beta and alpha plays this inhibitory, permissive gating role on the gamma beta that's carrying bottom-up information. So we think that this general circuit here is a modal cortical circuit that allows top-down information in your brain to control the processing of bottom-up information, allows you to control what you perceive, what you think about, and what you hold in memory. And with that, I thank you for your attention. And I thank all the people in my lab who actually do all the hard work conducting these studies. Thank you very much.
Thank you for your interesting and excellent presentation. And we have a couple of questions. The first one is, what is the neural substrate of deep layers operating at beta and superficial layers operating at gamma?
Sorry, could you one more time, please?
What could be the neural substrate of deep layers operating at beta, and superficial layers operating at gamma?
That's a great question. And So where beta and gamma comes from is a great question, and that work is best addressed by so far what we have is modeling work, and I turned you to the modeling work of my collaborator Nancy Capel. And basically what you get is you get these recurrent connections between neurons, and then clusters that represent the same stimulus. They're strongly recurrently connected, and they inhibit other clusters, other ensembles that represent another stimulus. And whether you get beta or gamma depends on the EI balance, the balancing excitatory and inhibitory influences in those circuits. And what's interesting, if you couple together these different ensembles, so you have them oscillating different frequencies, and you couple them together, you get this interesting phenomenon called traveling waves. These beta and gamma waves don't just stay in one place in cortex like a jump rope. They actually move across cortex when you connect these modules together. And so in some recent study we're just doing right now, we're finding that these oscillatory dynamics in cortex form traveling waves that move across the cortex in a task-dependent way.
Okay. So another question that occurs to me is how do you, if I understand correct, the increasing the beta oscillations is a kind of eraser of the memories already stored in the working memory in the working memory.
Sorry.
So my question is, what is the effect of this surge in beta oscillations into these layers presenting the beta oscillations? So kind of what is the mechanism that is inducing these oscillations?
Well, in the original Lundquist model, which we based our studies on, beta and gamma aren't actually separate circuits. They're different states of the same circuit. So a circuit is in beta when it's in a low energy state, when there's no bottom-up inputs. And then when you have a bottom-up input, the increased energy knocks the network up into gamma range, and that may explain this difference between this push-pull between beta and gamma, because they're two different states of the same network. So what you have is you have the spiking carrying bottom-up information that's going to tend to drive the superficial layers of cortex up into the gamma range. Then you have the beta carrying the top-down information that's has less energy. It's going to be in the beta range. As for the exact circuitry that allows beta to inhibit gamma, that is still a subject of investigation. We don't know the exact circuit, but it could be a matter of the beta dynamics switching this EI balance to turn on and off the expression of gamma and spiking. And one thing I'll mention is that for most of the talks, I didn't get to the predictive coding part, I've treated this beta signal, alpha beta signal, as if it's just a faucet turning on and off working memory. Our predictive coding study shows that it's not just this, it's much more precise than that. It actually targets the representation of specific stimuli. You can actually target, turn on and off the representation of individual stimuli using these beta gamma dynamics. So it's not a general faucet. It can actually be used to manipulate individual stimuli.
Thank you. And we have another question from the public. Jose, please, you can ask.
First of all, thank you, Errol, for your talk. Very, very good, too good. I have just one idea. And you started giving the example of visual cortex. Yep. And in visual cortex, you have this very classic and very well-known retinotopic representation. You show in your last part of the talk that may not be the case for working memory, at least in the prefrontal cortex, because you don't transfer in the experiments where you train the classifier, you don't get the same activity in one side and in the other side.
That's right.
Which would be expected for a nemotopic, if you can say, cortex, if the representation is exactly the same at one side of right. or the other, like more or less visual cortex regarding retina.
Right.
Two questions. It may seem the easy question is where do you think if we at some point of the building of memories we need this spatial coherence between the two sides or not? maybe not in the prefrontal cortex, but at other levels. And two, it may not be the case that activity, giving beta and gamma dancing, if you can say, is more important than the cells, the individual cells, at least in prefrontal cortex, not the case for visual cortex, V1.
Mm-hmm. Well, actually, could you unpack the last part of that question? What do you mean by the spikes, beta and gamma not being as important as spiking?
Meaning that it doesn't matter who is spiking, but the rhythm is which is in that particular cell engaged.
Well, I will say that, when we look for information about sensory information, for example, we don't find much information per se in gamma, in rhythms, in spike. Gamma seems to be just co-expressed with spiking, and we only can really decode information about the stimuli when we look at spiking. Okay. But alpha beta is a different story. Alpha beta, you can actually get top-down information. You can decode top-down information from the patterns of alpha beta. So there seems to be some difference there between alpha and beta can carry top-down information, but when it comes to sensory information, that's largely tied to spiking, but the spiking is associated with gamma. It helps traffic spiking. As for the first part of your question, it's true that we've known for a long time that prefrontal cortex is not retinotopic. But what we found in our studies, people sort of assume that maybe when you get to the level of prefrontal cortex, you got these huge bilateral receptive fields of neurons. So the neurons then bridge the gap between left and right. But that assumption was based on some very early studies in anesthetized animals. And what we learned is that the anesthetized brain is very different from the awake brain. And when we did these studies looking at the representation in the prefrontal cortex in the awake brain, there's no retinotopy, but there is a strong, heavy contralateral bias, just like you see back in visual cortex. I'm sorry, did that answer all your questions?
Yeah. What have you? I will make another question.
Okay.
Did you know what happened with monkeys or animals who have the corpus callosum, I don't know, slice or cut? Because where do you, anatomically, I'm thinking, where this transfer goes by?
Yeah, that is a great question. And we just now planning a series of experiments where we're going to record from all over cortex and look to see where this transfer is taking place. One possibility is it takes place in the prefrontal cortex. Then once a memory goes from one side of the brain to the other, and it gets fed back to the rest of the cortex. Another possibility is the transfer takes place at every level of cortex because there's connections to the corpus callosum at every level of the cortex. So the first thing we're going to do is we're going to do a neurophysiological survey of every level of cortex, prefrontal, parietal, visual cortex, different levels of visual cortex, and see where we see the evidence of this neurophysiological evidence for this transfer between the hemispheres. And then we'll have a better idea of what sort of manipulation experiments we can do to nail down where the transfer is taking place. I would hypothesize that I would guess that we're not going to see this, probably not an exclusive function of the prefrontal cortex. We'll probably see some active transfer even back in visual cortex. And in one of these studies, we're planning not just using working memory, but having the animal trace objects as they move back and forth between the right and left visual field. And we want to see if that gets handed off between the right and left visual field, not just in the PFC, but at different levels of cortex.
Sounds very, very cool experiments.
I hope so.
Yeah. Good luck with that.
Thank you.
Okay, we have another question from the public, and it's from Claudio Villalobos. He says, are beta and gamma oscillations independent or a correlated phenomenon? Will be reducing or deleting gamma will have any effect on beta oscillation?
Well, all over cortex, you see that beta or alpha, it's beta in frontal cortex, but alpha in visual cortex. And we've done a survey across cortex, and what you see is as you move from the front of the brain to the back of the brain, all the frequencies gradually drop down a little bit. So beta and alpha are pretty much the same thing, just in parts of the brain. All over cortex, you see beta, alpha, and gamma are anti-correlated. And the classic example is in motor cortex. You have the animal, or human for that matter, instruct them to make a motor response, but say, don't make it yet, hold on, wait for a go signal. You'll see lots of betas are suppressing the control signal, the motor response. And then as soon as they're giving the go signal, beta drops, gamma goes up. And there's also studies in visual attention. Whenever the animal, say the animal will attend to a stimulus, lots of gamma, and the animal will attend somewhere else, what goes on is alpha goes up for the to the place where you're not paying attention to. So these dynamics between alpha, beta, and gamma are anti-correlated all over cortex. So we think they're correlated in the sense that they're anti-correlated. They seem to be, if they're not inhibiting one another, they're at least different parts of the same, different states of the same network. So they're correlated in the sense that they're anti-correlated. You don't see many examples of, or any examples I know of, where higher frequency gamma and lower frequency alpha beta are actually coexistent. You do see cross frequency coupling where different frequencies will work together, but not for alpha, beta, and gamma. They tend to be really anti-correlated. You see coupling between like very low theta and gamma, so across a wider range, but not alpha, beta, and gamma per se. They seem to be, they really, everywhere you look, they seem to have this anti-correlated push-pull kind of relationship.
Okay, and the last question is that, there is any mechanism, or sorry, any task where the whole prefrontal cortex is engaged in this gamma or beta oscillations at the same time in both sides of the brain, not just one or two?
Oh, now that is a great question. That's another study we're doing. So I presented it as separate working memory source in the right and left sides of vision. And actually, my colleague Ed Vogel has studied 1,000 people now, and we find the same split in the human brain. That you really have, it really is as if we're all a bunch of split brain patients when it comes to visual working memory. Okay. Do we see any case where it's happening at the same time? Well, one example is what happens if I holding a stimulus of working memory that I look at directly at the center of vision, which is the way working memory has been studied for 50 years, or at least non-spatial working memory. You just look directly at a stimulus. What should happen there is the working memory should go to both hemispheres. So what happens? Are they coordinated between the hemispheres? Are they anti-correlated to the hemispheres? Are they independent? Nobody knows, because amazingly, we look back over the history of monkey neurophysiology, and no one has recorded bilaterally in the cortex. They've done what we probably what you guys have always done, what we always done. We record from one side of the brain and save the other side for a future experiment. So no one knows the answer to these questions because no one's ever recorded bilaterally in cortex. So just complete studies where we did the classic working memory study, just hold a picture that was presented at the fovea while we recorded bilaterally. So I literally should have an answer to you in about a month or so.
Okay, thank you for your really excellent talk. And okay, in honor of time, because we are late, we want to thank you for your willingness to present here. So thank you. Thank you, sir.
Thank you for the invitation. I really appreciate it.
Okay, so for tomorrow we have 4 more presentations. So we will start at 9, and the link will be sent to everybody tomorrow at 8:45. Thank you so much.
Bye.
Thank you. Bye.
Bye. Thank you. Thank you all.
Bye.
Bye. Bye.
Bueno, Antonello. Ahí estáis de... Ya, nos vamos. Un abrazo. Salió súper bien. Cuidarse. Gracias, Pablito. Un abrazo.
Oye, Pepe, ¿te ha afectado en la pandemia, parece?
Espérate.
Oye, estamos en YouTube. Así que...